From just below the surface of the ocean, on a calm day, the light moves in long shifting patterns across the water above. It bends and refracts with each small ripple at the surface, spreading itself into a slow-moving lattice of pale blue and white that drifts downward through the first few meters of open water. The color is specific and distinct, not the blue of a painted wall or a clear sky, but something with more depth to it, more internal, as if the light is being gently filtered through the water itself rather than merely traveling through it. Standing at the surface and looking down, the eye follows this column of illuminated water until it fades, which it does by degrees, darkening from turquoise to a deeper blue to something closer to indigo, until finally it passes beyond what any amount of looking can resolve. In the deepest places on Earth, that column continues for nearly 11 kilometers beneath where the light gives out. The ocean is not a single environment. It is a series of layered worlds, stacked one above the other, each with its own temperature, pressure, and population of living things, separated by invisible boundaries that may shift with the seasons but rarely dissolve. The uppermost of these worlds is the one most familiar to anyone who has swum in the sea or looked down from a boat into clear water, a sun-warmed layer stretching from the surface to roughly 200 meters depth, where enough light filters through to support photosynthesis. This is the epipelagic zone. It is the only part of the ocean that most people will ever inhabit, even in imagination. Below it, the water changes in ways that are not immediately visible from the surface, but are felt by anything moving through them, a gradual and then more rapid departure from warmth and light, and the possibility of photosynthesis into something quieter and more ordered and altogether different. Between approximately 200 and 1,000 meters depth, the temperature drops in a way that is not gradual but steepens. The surface ocean, warmed by sunlight, sits above this boundary as a body of relatively warm and comparatively light water. Below it, the colder water that has never been touched by direct sunlight is denser, heavier, more reluctant to move. The transition between the two, the layer where temperature falls most sharply with each additional meter of depth, is called the thermocline. A diver descending through it does not cross a sharp line, but passes through a gradient, a few dozen or few hundred meters of water, where each meter cooler than the last, until the temperature stabilizes at the cold, even conditions that define the deep ocean. If a hand were extended downward at the upper boundary of the thermocline, the water below the fingertips would be noticeably cooler than the water at the wrist. The thermocline holds these two bodies of water apart, not through any active process, but through density alone, the cold, heavy water below and the warm, lighter water above, each staying where physics keeps it, the boundary between them persisting for months at a time. Below the thermocline, in the mesopelagic zone, stretching from 200 to 1,000 meters, the water holds its cold with consistency. The light that penetrates this far is minimal, less than 1% of what falls on the surface reaches even the upper boundary of this zone, and what remains at the lower boundary is less than that, a last trace of filtered blue that the human eye would barely register. The creatures living here have adapted to this specific quality of near darkness, and their bodies reflect the conditions, eyes enlarged beyond what a surface animal would need, bodies often small and dark or silver, oriented to the faint glow from above. Life in the mesopelagic zone is defined by this specific relation to the fading light, a community of organisms organized around the last of it. The thermocline is also a kind of barrier to exchange. Surface water, warmed and stirred by wind and sun, carries dissolved gases and nutrients in one direction. Cold, deep water, dense and still, carries its own particular chemistry in another. Because the warm water floats and the cold water sinks, and because the thermocline holds them apart, the two layers do not freely mix. Nutrients that fall from the surface as dead organic matter do not easily return upward. The mixing that does occur happens slowly, driven by storms, large-scale current systems, and the gradual working of tidal forces against continental shelves. The thermocline is not a wall, it is a slowing, 1,000 meters. At this depth, the last of the surface light is gone, not diminished to a faint trace, but genuinely absent. There is no wavelength of sunlight that reaches this deep under ordinary ocean conditions. The temperature has settled at a few degrees above freezing. The pressure, which increases by approximately 1 atmosphere for every 10 meters of depth, has now reached roughly 100 times the pressure at the ocean surface. A creature living at this depth and carrying an air-filled cavity in its body, a swim bladder, a lung, any internal space not filled with fluid, would find that cavity compressed by the surrounding water to a fraction of its surface dimensions. The animals that do live here have no such cavities. Their bodies are built from materials that compress as readily as water itself, flexible cell membranes, fluid-filled tissues, bodies that offer no rigid resistance to the pressure because they have become, in a sense, continuous with it. They do not experience the weight of 100 atmospheres as a crushing force. They are permeated by it. The zone below 1,000 meters and above 4,000 is called the bathypelagic zone. There is no other name for it that captures what it is as precisely as that. The zone of the deep. It contains none of the features that make the upper ocean feel dynamic. No light, no photosynthesis, no seasons in any surface sense, no warmth from above. Temperatures in the bathypelagic zone sit between approximately 2 and 4 degrees Celsius across most of the world's ocean basins. They do not vary much with depth, and they do not vary much over time. A column of water at 3,000 meters in the North Atlantic holds roughly the same temperature in January as in July, and roughly the same temperature this year as it did a century ago. This stability is one of the zone's defining qualities. There is an exceptional stillness here that accumulates rather than settles, not the stillness of a room where someone has just left, but the stillness of a place that has not been disturbed in the ordinary sense for a very long time. The creatures of the bathypelagic zone have adapted to this stillness. Among them are anglerfishes, which carry bioluminescent lures above their heads and wait in the dark for smaller animals to investigate. There are black swallowers, small fish with enormously distensible stomachs, capable of engulfing prey several times their own body size. Deep-sea jellyfish move through the bathypelagic water in shapes that would seem alien at the surface. Flat bells, long trailing filaments, transparent bodies through which the dark water is dimly visible. These are not accidental inhabitants of the zone. Each one is a product of the same long process that placed every animal precisely in the conditions that shaped it, and the bathypelagic zone, for all its unfamiliarity, is as coherent an environment as a temperate forest or a coral reef. Its inhabitants know it completely, in the sense that all wild animals know their habitat, not consciously, but in every detail of their physiology. The stillness at 3,000 meters is of a specific quality. There is no weather here. No storm on the surface communicates itself to water at this depth in any form the water would recognize as a storm. Wind that drives 5-meter swells on the surface has no direct influence at 3,000 meters, where the water sits in its enormous cold column, undisturbed. What does reach this depth from above? A slow rain of organic particles, the long-period waves of tidal forces, the faint echo of large earthquakes, arrives transformed, attenuated, nothing like its source. A column of near-freezing dark water at 3,000 meters can sit undisturbed for months at a stretch, moved only by the extremely slow deep ocean currents that carry water across ocean basins over the course of centuries. It is not inert, but it is patient in a way that has no analogy in the surface world. Below 4,000 meters, the character of the ocean changes again. The abyssopelagic zone, stretching from 4,000 to 6,000 meters depth, represents the deepest open water environment before the ocean floor's irregular topography begins to intrude. Most of the world's ocean floor lies within this depth range, vast flat plains of sediment, punctuated by seamounts and ridges, but largely level, covered in the slow accumulation of particles that have drifted down from above over millions of years. The water here is only a degree or two above freezing. Pressure has reached 400 to 600 atmospheres. The oxygen content of the water, which drops through a minimum zone in the mesopelagic layer, has by this depth stabilized to some extent, and life continues here, though at densities far lower than the surface. Fish, crustaceans, and invertebrates of various kinds inhabit the abyssopelagic zone, most of them adapted to its particular combination of darkness, cold, and pressure with the same thoroughness as their counterparts in the zones above. 5 zones. 5 distinct environments stacked in a column that, in most parts of the ocean, has no bottom for kilometers. The deepest water column in the world is not in the abyssopelagic zone at all, but in the 6th and deepest environment, the Hadal zone, named for the ancient Greek concept of the underworld, which occupies the trenches descending below 6,000 meters. Trenches are not merely the deep parts of the ocean floor. They are a distinct geological feature. Narrow, elongated valleys in the ocean floor formed at subduction zones, where one tectonic plate slides beneath another, pulling the seafloor downward into a steep-walled trench that can extend several additional kilometers below the surrounding abyssal plain. From above, a trench looks on a map like a long, narrow scar in the ocean floor. In cross-section, it is more like a valley, not the wide, gradual kind, but the steep-sided kind, walls descending at angles that accumulate sediment in particular ways and enclose a community of organisms in conditions that differ even from the abyssal plain, a few kilometers shallower. There are approximately 27 known trenches worldwide. They are not evenly distributed across the ocean floor, but clustered along the active margins of the Pacific, where tectonic subduction is most pronounced, with others in the Indian Ocean and elsewhere. Their names in some cases reflect the geographic features they adjoin: the Tonga Trench in the southwestern Pacific, the Philippine Trench in the western Pacific, the Kermadec Trench northeast of New Zealand, and the largest and deepest of them, the one whose name has become almost synonymous with the concept of ocean depth itself, the Mariana Trench, located in the western Pacific Ocean, east of the Mariana Islands, home to the Challenger Deep, the deepest known point in any ocean on Earth. The Mariana Trench descends to approximately 11 kilometers at its deepest point. This number is worth dwelling on for a moment, not because it is dramatic in its implications, but because it is difficult to hold in mind as a physical fact. 11 kilometers is roughly the cruising altitude of a commercial aircraft. A person standing at the ocean surface above the Challenger Deep and looking downward would be looking through a column of water roughly equivalent in the vertical dimension to the distance between the ground and the clouds that form the upper boundary of most weather systems. The ocean, at that point, is deeper than the sky above it, is high, Most of the water in that column is cold, dark, and still, sitting undisturbed in the specific conditions of whatever zone it occupies, with no awareness that above it or below it any other condition exists. The Hadal Zone is defined as the environment below 6,000 meters, the zone found only in trenches, not on the open abyssal plain. Its inhabitants include creatures that have adapted specifically to the conditions found in these narrow, deep valleys. Extreme pressure, total darkness, very cold temperatures, and a somewhat unusual food supply. Because trenches are concave features in the ocean floor, organic matter drifting down from above tends to accumulate there. The rain of particles that falls through the water column above, dead organisms, feces, fragments of organic material, gathers in the trench floor in concentrations higher than on the surrounding plain. The Hadal Zone is, in a certain sense, a collection point. What drifts down from the 5 zones above eventually settles here, in the deepest valleys of the ocean, where it becomes the food supply for the communities of organisms that have made this extreme environment their own. Among those organisms are the amphipods, small crustaceans, shrimp-like in form, that thrive in Hadal trenches in extraordinary densities for so deep an environment. They have been found in virtually every trench that has been sampled, often in numbers that surprised the scientists who collected them. An animal that flourishes at 11,000 meters depth must be physiologically unlike its shallow water relatives in significant ways. Its cell membranes, its enzymes, its general biochemistry must all function under pressures that would disrupt the molecular architecture of a surface-dwelling organism. The adaptations are specific and numerous, and they are not recent improvisations. The ancestors of Hadal amphipods have been working out the details of deep trench survival for tens of millions of years. Snailfish have also been found in Hadal trenches, small, translucent, eel-shaped fish whose soft bodies and absence of rigid air-filled cavities allow them to function at depths that would be impossible for animals built on a more rigid plan. Observations from crude and robotic vehicles have recorded snailfish at depths exceeding 8,000 meters in several trenches. They move with a quality of ease that is particular to their body plan. Not swimming exactly, not hovering, but drifting and adjusting in the dark water with a casual efficiency that suggests they are entirely at home in the conditions around them. They are. The deep trenches, which seem from the outside to be places defined by extremity and inhospitality, are from the inside simply where these animals live. Between the trenches, connecting them in the deepest sense, is the abyssal plain, the wide, cold, dark floor of the ocean that constitutes most of the seafloor's area. This is not a dramatic landscape in any conventional sense. It is flat, or nearly so, in most places. It is covered in sediment that has been building up for millions of years. It is cold and dark and under enormous pressure, but it is also inhabited, continuously and in many ways, by creatures that have been living there since long before any human looked into it. The abyssal plain is not waiting for something to happen. It is a functioning ecosystem, running on the same slow currency of energy that has always sustained it, at the same temperatures and pressures, in the same darkness. Returning for a moment to the surface, to that column of illuminated water in the epipelagic zone, where light moves in shifting patterns through the first 200 meters, and the temperature is warm enough to sustain the photosynthesis that drives most of the ocean's biological production. The connection between this lit, warm, productive surface layer and the cold, dark, enormous volume of water beneath it is not a coincidence of geography. It is an active relationship. What happens at the surface determines what falls through the water column. what accumulates in the deep zones, what arrives at the trench floors after its long descent. The surface and the deep are linked by gravity and time, not quickly, not dramatically, but continuously and without interruption. The five zones, taken together, occupy a volume of water that dwarfs any other habitat on Earth. The total volume of the world's oceans is approximately 335 billion cubic kilometers. Of this, roughly 95% lies below 200 meters, below the epipelagic zone, below the reach of meaningful sunlight, in the mesopelagic and deeper zones where most of the ocean's biological and chemical work takes place out of reach of the surface. The surface ocean, the part that most people mean when they think of the sea, is the thin, lit, warm outer edge of something immensely larger. It is the visible part of a system that extends, in some places, to depths that take the imagination several additional attempts to reach. The mesopelagic zone alone, the twilight layer between 200 and 1,000 meters, has a volume so large that estimates of the total biomass it contains have been revised significantly upward in recent decades, only as sampling methods improved and the density of life in that zone turned out to be considerably higher than older techniques had suggested. The deep ocean contains life not as a sparse, scattered presence in an otherwise empty dark, but as a continuous and in some ways remarkably dense community of organisms occupying every depth and every part of the water column, each adapted to the specific conditions of its zone. The bathypelagic zone's particular quality of stillness deserves a closer look. At 3,000 meters depth, the water does not merely lack light and warmth. It lacks the turbulence that characterizes the surface, the mixing, the vertical transport, the horizontal current variability driven by wind. The currents that do move water at bathypelagic depths are driven not by wind, but by density differences, and they are extraordinarily slow by surface standards. Water at 3,000 meters in the North Atlantic may be moving horizontally at speeds measured in centimeters per year. A parcel of water at that depth can sit in roughly the same position for years, decades, cooling gradually, carrying its dissolved load of oxygen and carbon dioxide and nutrients on a journey that will eventually, over centuries, bring it back to a place where it can exchange with the surface again. The stillness is real, but it is not permanent. It is simply patient. The pressure at the base of the bathypelagic zone, 4,000 meters depth, is approximately 400 atmospheres. This is 400 times the pressure at the ocean surface, or the equivalent of roughly 400 kilograms pressing down on every square centimeter of whatever surface is at that depth. For a rigid container with an airspace inside, this pressure would be catastrophic, an implosion happening faster than any perception could register. For the animals living at that depth, none of this is a problem. They have no air spaces. Their bodies contain no gas-filled cavities. Their cell membranes are built from lipids with a particular flexibility. At this pressure, their proteins fold and function correctly under conditions that would denature the equivalent proteins in a surface animal, and the pressure itself passes through them as it passes through the surrounding water without destroying them. Adaptation to pressure over the enormous spans of time in which these animals evolved is not a workaround, it is a design. The abyssopelagic zone, sitting between 4,000 and 6,000 meters, represents the deepest open water column before the trench floors. It is dark, cold, and under pressure that ranges from 400 to 600 atmospheres, depending on exact depth. The few fish and invertebrates inhabiting this zone are among the least studied animals on Earth, not because they are rare in the sense of existing in small numbers, but because reaching them with sampling equipment is technically demanding and expensive. What is known of them is largely derived from the specimens that have been captured in deep sea trawls over the past century and a half of oceanographic research. Though in recent decades, remotely operated vehicles have begun to extend the catalog of observations to living animals in their actual habitat. The picture that emerges from this work is not one of a zone struggling to support life, but one of a zone that has been supporting life in its own terms and on its own timetable for a very long time. Below 6,000 meters, the trench walls close in. The Haddle Zone is not a single environment, but a collection of isolated ones, each trench its own enclosed deep, separated from other trenches by thousands of kilometers of abyssal plain. Its Haddle community more or less cut off from the Haddle communities of other trenches. This isolation has biological consequences. Species found in one trench are not necessarily found in others, and the populations within individual trenches are genetically distinct from their relatives in adjacent trenches in ways that reflect their long separation. The Tonga Trench and the Kermadec Trench, though relatively close to each other in Pacific terms, have different Haddle communities, communities that have been evolving in isolation long enough to diverge in measurable ways. The trench floor is not just an extreme environment. It is a set of isolated extreme environments, each with its own evolutionary history. The 5 zones of the open ocean, epipelagic, mesopelagic, bathypelagic, abyssopelagic, hadopelagic, are not equal in thickness. The epipelagic zone, at 200 meters, is thinner by far than any of the others. The mesopelagic zone is 800 meters thick. The bathypelagic spans 3,000 meters. The abyssopelagic, another 2,000. And the hadopelagic extends to whatever the maximum depth of the deepest trench happens to be, a further 5 kilometers below the base of the abyssal pelagic. When these depths are laid out as proportions of the total water column in the deepest ocean, the epipelagic zone where all surface photosynthesis takes place is less than 2% of the total depth. The rest is dark. The thermocline, sitting between the epipelagic zone and the mesopelagic, is a boundary of exceptional significance. It is not merely a thermal gradient, it is a barrier to the movement of nutrients, oxygen, and organisms between the warm surface ocean and the cold deep ocean. Its depth and intensity vary with latitude and season. In tropical waters, the thermocline is permanent, strong, and positioned at relatively shallow depths. In polar waters, it weakens or disappears, allowing surface and deep water to mix freely. These regional differences have consequences for the productivity of surface ecosystems. Where the thermocline is strong and permanent, the surface water tends to run low on nutrients, because the nutrients that fall downward do not return. Where the thermocline is absent or weak, nutrients can cycle back to the surface more freely, and productivity is accordingly higher. The thermocline is, in this sense, a dial. Where it is strong, the surface ocean is clear and beautiful and relatively sparse. Where it is weak, the surface ocean is often greener, richer, more visibly alive. The naming of the ocean zones has a practical history. The word pelagic comes from the Greek for the open sea, and the prefixes that differentiate the zones, epi-high, meso, bathy, abysso, haddle, describe their position in the water column from above. Epipelagic, meaning upon the sea. Mesopelagic, meaning middle. Bathypelagic, from the Greek for deep. Abyssal pelagic, from the word for the bottomless. And hadopelagic for the underworld. The Greek underworld, Hades, carried into the naming of a zone that descends beneath all the others into the deepest places accessible on Earth. There is something apt about this vocabulary. The zones were named by people who were trying to bring order to something they could not directly observe, working with what depth-sounding equipment and sampling technology could tell them, mapping a space that resisted conventional mapping. The words they chose reflect the scale of the attempt. The Hadal Trenches, when considered individually, are each a specific geographic and geological feature with their own histories, dimensions, and populations. The Mariana Trench reaches approximately 2,500 kilometers in length and about 69 kilometers in width, a narrow feature relative to its length, running roughly northeast-southwest in the western Pacific. The Tonga Trench descends to approximately 10,800 meters and is considered among the most seismically active trench systems in the Pacific. The Philippine Trench reaches depths of over 10,000 meters and runs along the eastern edge of the Philippine Islands. The Kermadec Trench, northeast of New Zealand, descends to approximately 10,000 kilometers and has been the site of numerous deep-sea biological surveys in recent decades, producing important records of hadal organisms, including the amphipods and snailfish that represent the best-studied components of hadal community ecology. Each of these trenches descends below 6,000 meters into its own isolated world. The water inside a trench is not meaningfully different in chemical composition from the water on the surrounding abyssal plain, but it accumulates organic matter faster. It holds isolated populations of organisms that have evolved in seclusion, and it represents a depth that is, in practical terms, as remote as any place on Earth. The instruments that have visited these depths can be counted. The systematic biological surveys that have been conducted in them are more limited still. What is known of hadal communities comes from a relatively small number of samples and observations spread across the decades since the first dedicated hadal research began in the mid-20th century. But what those samples and observations have established is clear enough. The hadal trenches are not empty. They are inhabited by communities of organisms adapted to their specific conditions, with the same thoroughness and the same long evolutionary history as the organisms of any other zone. The amphipods of the Mariana Trench, the snailfish of the Kermadec, the specific microbial communities that colonize the sediment at the base of each trench, all of these are living in the hadal zone right now, in the dark and the cold and the pressure, going about the business of feeding and reproducing and dying in a place that receives no sunlight and almost no human attention. The ocean at its surface, on a calm day, is a specific blue. This specific quality of the surface color, something between turquoise and deep blue, something that shifts with depth and cloud cover and the angle of the sun, is only possible because the water is clear enough that light can enter it and be scattered back. Below the first 200 meters, that clarity continues, but the light that generated the color at the surface does not. By the time the water column reaches 1,000 meters, the light is gone. By 3,000 meters, the temperature is near freezing and the pressure is 300 atmospheres, and the water has been sitting in the dark for a very long time. By 6,000 meters, the abyssal plain has given way to the trench floor, and the walls have closed in to create an environment that is isolated from the plain above it, in the way that a valley is isolated from the plateau, not completely, but enough. At the bottom of the Mariana Trench, the Challenger Deep, nearly 11 kilometers below the surface, the water is cold and dark and still. The sediment on the floor is fine, soft, pale. The pressure is over 1,000 atmospheres. There is no light from above, no current from any weather system at the surface, no warmth from any sun. There is, by all accounts, life, microbial life in the sediment, amphipods in the water column above the floor, possibly other organisms not yet cataloged. And beyond the Challenger Deep, running in long, narrow valleys across the floor of the Pacific and the Indian Ocean and elsewhere, the other trenches continue their own existence. The Tonga, the Kermadec, the Philippine, and the roughly 23 others that descend below 6,000 meters into their own isolated floors. Each one is sitting in stillness right now, far beneath any ocean any of us can see from where we are, holding its own cold, dark water, its own thin sediment, its own small communities of adapted life, in the deepest and most patient part of the layered world that began not so far above in the shifting blue light at the surface. Somewhere near the surface of a clear ocean, on a day with no wind, sunlight enters the water and immediately begins to change. The change begins within the first meter. Red light, the longest wavelength in the visible spectrum, does not travel far through seawater. It scatters and is absorbed almost at once, so that within a meter or two of the surface, the warmth that red carries with it has quietly left the water column. Not suddenly, not with any visible event. The water simply no longer holds it. One moment there is a full spectrum of light entering from above, and then there is slightly less of it, and the part that is gone is the warm end, the part that corresponds to fire and earth and late afternoon. It drains out of the water the way warmth leaves a stone after the sun goes behind a cloud, gradually, without announcement, and without return. Orange follows, then yellow. Green holds on longer than most, persisting through tens of meters before it too thins and disperses, and what remains below it is a single color, blue. Deep, even directionless blue, growing steadily dimmer as the depth increases, but holding its character all the way down through the upper ocean, the one reliable wavelength in a column of water that has filtered everything else away. By around 200 meters, even this last color has reduced to less than 1% of the light that entered at the surface. Below that point, the ocean absorbs what little remains. The sun, which covers more than half the sky from the surface, becomes irrelevant. This is not darkness the way a room is dark when the lights are off. It is a graduated physical process, the ocean doing what water does to light, wavelength by wavelength, with the longer ones going first and the shorter ones lasting longer, the whole spectrum unwound and removed layer by layer on the way down. By the time the water is truly dark, it has been working on the light for a long time, and the light has given up everything it had. The zone between 200 and 1,000 meters does not lose its light all at once. In the uppermost part of it, around 200 meters, the water is still faintly bright, not enough to read by, not enough to cast a shadow, but enough that a sensitive eye might detect it. The light here is blue, filtered down from the surface, and it arrives from above as a dim and diffuse glow rather than as rays. Nothing casts a shadow. The illumination is everywhere and nowhere, a luminous haze that fades evenly in every direction, so that up is slightly brighter than down, and everything else is the same dim blue. This is the twilight zone, the mesopelagic region, and the creatures that live here have had a very long time to make use of the small amount of light it offers. A lanternfish at 300 meters is a small animal. Most species fit easily in a closed hand. The body is silver, which is common among open ocean fish because silver reflects the ambient color of the surrounding water and makes the fish difficult to see from any direction. But the lanternfish has something else, which is a set of small light-producing organs along its underside, photophores, each one a tiny self-contained point of light. Seen from above, the fish is a silver shape against a dim blue background. Seen from below, Looking upward toward the faint surface glow, the fish would cast a dark silhouette, except that the photophores on its belly produce a faint blue-green light that matches closely enough the dim downwelling light from above. The silhouette dissolves. The fish becomes the water around it. This is called counter-illumination, and the lanternfish is not the only animal that uses it. Hatchet fishes do the same thing, so do certain squid. The logic is the same in each case. In a zone where some light still arrives from above, the most dangerous angle of visibility is from below, looking up into the brighter water. By emitting light from the underside to match what comes from above, an animal can erase its own outline and become, in effect, continuous with the column of water it swims in. The predators below it, looking upward, find nothing to distinguish it from the faint blue glow overhead. The hatchetfish carries this strategy to an extreme. Its body is deep and flat, like a hatchet seen from the front, all of the volume compressed into a narrow vertical profile, so that the surface area facing downward, the surface that needs to be lit, is minimized. The eyes of a hatchetfish are proportionally very large, and they point upward rather than sideways, oriented toward the dim light from above, because that is where anything of interest is most likely to be silhouetted. The fish itself is nearly invisible from below. What it watches for are the outlines of other fish backlit against the surface glow. The bristlemouth is less often described than the lanternfish or the hatchetfish, but it may be more numerous than either, more numerous, in fact, than almost any other vertebrate on Earth. The various species of bristlemouth fish, small, dark, slim, with rows of tiny sharp teeth that give the group its name, are distributed across the mesopelagic zone in such quantities that some estimates suggest their collective numbers rival or exceed those of any other group of vertebrates. Every ocean, at the right depths, holds them in the billions. A trawl net dragged through the twilight zone brings up more bristlemouths than almost anything else. They are the common matter of that layer, the fish that fills the volume the way small songbirds fill a temperate forest, invisibly and in enormous numbers, doing what small predators do in a large, food-limited environment. They too have large eyes for their size, oriented upward in the characteristic way of mesopelagic fish, capturing the last faint traces of downwelling light from above. They too are dark on their upper surfaces and lighter below. They migrate vertically each day, rising toward the surface at night, when darkness removes the risk of being silhouetted, descending again at dawn to sit in the dimmer twilight zone through the day. This daily migration, up and down through hundreds of meters, is one of the largest coordinated movements of animal mass on the planet, happening every day in every ocean, largely invisible because it happens in open water far from any coast. Below 1,000 meters, the last trace of surface light disappears entirely. The word for this threshold, the depth at which the sun finally and completely lets go, is not dramatic in the way the boundary feels when described. It is just a line on a diagram, a number. But below it, the conditions change in a way that no amount of visual adaptation can bridge. Eyes tuned to faint blue light find nothing to work with. There is no downwelling glow to match, no overhead brightness to watch for silhouettes against. The water below 1,000 meters is, in the most literal physical sense, absent of any light from the surface. What light exists below that line has been made by living things. The anglerfish is one of the better-known inhabitants of the deep ocean, and its light-making organ, called the esca, is worth describing carefully because it works in an unusual way. The esca is a small bulbous structure attached to the end of a modified dorsal spine that extends forward above the fish's head like a fishing rod extended ahead of it. The light within the esca does not come from the fish itself. It comes from bacteria, dense colonies of bioluminescent bacteria living symbiotically within the tissue of the lure, provided with nutrients by the fish and producing in return a continuous blue-white glow. The fish cannot turn this light off and on the way some bioluminescent animals can. The esca glows continuously, a small point of light suspended in total darkness, produced not by any deliberate act of the fish, but by the steady biochemical activity of the organisms living inside it. In the complete dark of the bathypelagic zone, this light is visible from some distance. An anglerfish hangs nearly motionless in the water, jaw open, lure extended forward, the esca glowing above its mouth like a lantern held by a very still figure in a very dark room. Other animals, smaller fish, crustaceans, whatever is drawn to light in a place where light does not naturally occur, are attracted to it. They approach. The fish closes its jaw. The sequence is patient and economical, requiring almost no expenditure of energy from the anglerfish beyond the metabolic cost of waiting. The light in the esca is produced by a biochemical process that begins with a molecule called luciferin. An enzyme, luciferase, catalyzes the reaction, introducing oxygen and producing an excited molecule that releases its extra energy as light rather than as heat. The efficiency of this process is genuinely notable. Nearly all of the energy put into the reaction comes out as light, with almost no heat produced as a byproduct. A comparable amount of energy run through an incandescent bulb would produce far more heat than light. The bioluminescent reaction produces the reverse, almost pure light, cool and steady, exactly the color that travels farthest through seawater. This biochemical pathway, luciferin, luciferase, oxygen, light, appears across the deep ocean in species with no close relationship to each other. Dinoflagellates use it. Jellyfish use variations of it. Squid use it. Certain fish produce their own luciferin rather than hosting bacteria that do. The molecules differ in detail across these lineages, and the structures that house them differ enormously, from a simple skin cell to an elaborate focusing lens and reflector, but the underlying chemistry is recognizably similar. This is convergent evolution, the same general solution arrived at independently many times across the long history of deep ocean life, because that particular solution, cool light, blue-green wavelengths, efficient and continuous, works particularly well in the environment where it evolved. Lanternfishes produce their own light rather than hosting bacteria. The photophores along their undersides are made of specialized cells containing luciferin, organized into small organs with reflective backing and sometimes with focusing structures that direct the light downward in a controlled way. The same fish that uses these photophores for counter-illumination during the day may also use them for other purposes, signaling between individuals of the same species, recognition of mates, coordination within a school. In the twilight zone, where the ambient light is just enough to see shapes against, a pattern of photophores along the flank of a fish communicates information that a fish at depth cannot get from color, because color has already been filtered out. The pattern itself, its arrangement, its timing, its intensity becomes the signal. Deeper in the water column, in the zone where the last blue light has faded, the variety of bioluminescent strategies increases rather than decreasing. This seems counterintuitive. If there is no ambient light to compete with, any light would be visible, but the darkness creates its own pressures. An animal that glows continuously is visible to every predator in range. An animal that produces light in short pulses or only when disturbed, or only from specific angles is harder to locate. Some animals produce bright flashes to startle and confuse predators. Some release clouds of bioluminescent mucus that distract while the animal retreats into the dark. Some use light to communicate in ways that only members of their own species can interpret. The siphonophores, colonial animals that drift through the deep water as long chains of specialized units, produce bioluminescence in some of their zooids. The night. A siphonophore trailing through the midnight zone may have photophore-bearing members positioned at intervals along its length, so that when disturbed, the whole structure lights up at once in a long, startling pulse. Seen from a distance in the dark, this might resemble the bioluminescence of a larger animal, a confusion that could give a predator pause. Whether this is an evolved strategy or a consequence of the colony's architecture is a question that has not been fully resolved. Deep-sea jellyfish produce bioluminescent light in their bells and tentacles. Some species, when touched or disturbed, produce cascades of blue-green light that move across the bell in waves, a physical response to mechanical disturbance that is probably not directed at any particular outcome, but that nonetheless makes the animal briefly and dramatically visible in the dark water. There are jellyfish in the bathypelagic zone whose bells are transparent except for the bioluminescent structures running through them, so that when they light up, they appear as geometric patterns hanging in the dark rings, spirals, grids of cold light, before the light fades and the animal becomes invisible again. What all of this light has in common is its color. Almost all deep-sea bioluminescence is blue-green, peaking around 470 to 490 nanometers on the electromagnetic spectrum. This is not a coincidence. Blue light travels farther through seawater than any other wavelength. The ocean absorbs red, orange, yellow, and green before they have gone very deep, but blue persists. An animal that produces red light in the deep ocean is producing a signal that attenuates quickly, visible only at short range, while an animal producing blue-green light is producing a signal that can travel much farther through the water before being absorbed. For most deep-sea organisms, blue-green light is simply the efficient choice. It carries the most information the farthest distance in the medium they inhabit. One fish has found a use for the exception to this rule. The dragonfish carries a bioluminescent organ beneath each eye that produces red light, not blue-green, red, the wavelength that ocean water absorbs most readily, the color that disappears within the first meter or two of the surface, and is essentially absent from the deep ocean under any natural conditions. Most deep-sea animals have no photoreceptors sensitive to red light. Their eyes are tuned to blue because blue is what their world contains. A dragonfish swimming through the midnight zone with its red photophores active is for most of its neighbors invisible, not because the light is too faint to see, but because the light is in a wavelength they have no capacity to detect. The dragonfish, however, can see red. It has photoreceptors sensitive to the wavelengths it produces, so it moves through the dark with a beam of red light extended forward, illuminating the water ahead in a color that is, to everything except the dragonfish itself, not there. Prey animals in its path, small fish, crustaceans, whatever, drifts in its range, are lit up by a light they cannot see, observed by an eye they cannot know is watching, approached by something moving toward them through what is, from their perspective, unchanged and total darkness. This is an unusual evolutionary development, because the production of photoreceptors sensitive to red light in a deep-sea environment requires both a source of red light and a reason to see it, and these two things have to evolve together or neither is useful. The working hypothesis is that the dragonfish's red light system evolved as a private communication channel, a way to signal to other dragonfish without those signals being visible to prey or to competing predators. Whether the prey illumination use came later, or whether both uses developed in parallel, remains an open question. The dragonfish is not common enough, and the deep ocean is not easy enough to observe, for the natural history of this particular adaptation to be fully written. There are other deep-sea animals that produce red bioluminescence, and artists and researchers have suggested that the phenomenon may be more widespread in the deep ocean than was previously assumed, that more animals than currently known may have both the capacity to produce red light and the photoreceptors to see it, running private channels of communication and illumination through a dark that to blue sensitive eyes appears empty and complete. The evidence for this is preliminary, and the question is not settled, but it adds a particular quality to the picture of the deep ocean's darkness, even in the place where all surface light has been gone for 1,000 meters. The darkness is not the same to everyone in it. A different kind of light appears at the very surface of the ocean, where the water is warm and the surface is disturbed by wind. Dinoflagellates, single-celled organisms that drift in enormous numbers through the upper ocean, produce brief flashes of blue light when the water around them is agitated. A breaking wave, a boat's bow, the passage of a fish through a dense bloom, any mechanical disturbance triggers a flash from the cells nearby, which triggers more flashes from cells adjacent to them, the light spreading outward from the point of disturbance in a ripple of cold blue fire that fades within a second. The light is made by the same biochemical process as the anglerfish's escargot and the dragonfish's photophore, luciferin, luciferase, oxygen, but in a cell so small it is invisible to the naked eye, producing a flash so brief it takes specialized instruments to measure its duration. On a dark night, a sea dense with dinoflagellates can produce visible light with any disturbance of the water. The bow wave of a ship glows blue. Fish moving beneath the surface leave trails of blue light behind them. A hand drawn through the water trails luminescence from every finger. The light fades almost immediately, but while it lasts, it is genuine and visible, a product of chemistry happening in millions of cells responding to the same mechanical event, each one doing what its chemistry instructs, with no more coordination or intention than a set of dominoes falling in sequence. This surface light and the light of the anglerfish's esca are in a meaningful sense the same process. Luciferin, oxidized by luciferase, releasing photons in the blue-green range, requiring oxygen, producing negligible heat. What separates them is not chemistry but context. One happens in warm, sunlit water at the surface, triggered by physical disturbance, lasting less than a second, produced by an organism too small to see. The other happens in cold, lightless water, a kilometer down, glowing continuously, produced by bacteria living inside a larger animal that has evolved an elaborate structure to house them and present their light to the world around it. The mesopelagic zone that holds the lanternfish and the hatchetfish and the bristlemouth is physically a layer defined by depth and by the amount of light it receives, but it is also defined by pressure. At 500 meters, the pressure on any object is about 50 times the pressure at the surface. At 1,000 meters, it is roughly 100 times. The organisms that live here have no air pockets, no swim bladders holding gas that would compress and collapse under such pressure, or if they do, they are specially adapted to manage it. The bodies of mesopelagic fish are often softer and more waterlogged than surface fish, their tissues having a higher water content and greater flexibility, their cell membranes tuned to remain functional at pressures that would alter the behavior of fats and proteins built for surface conditions. A fish adapted to 600 meters does not survive being brought rapidly to the surface. The pressure change is too great, too fast, and the body built for depth cannot reorganize itself quickly enough to function at the low pressure of the surface environment. This means that many of what is known about mesopelagic and deeper organisms comes from specimens that did not survive the journey to the surface, arriving at the deck of a research vessel in a state that reflects the trauma of rapid decompression rather than the normal state of the living animal. This is one of the reasons that filmed observations, cameras lowered to depth, remaining at depth, have added so much to the picture of deep ocean life. The dragonfish in its natural habitat, moving through the midnight zone with its red beam active, does not look like a dragonfish trawled to the surface and pulled from a net. The blue-green color of most bioluminescence is, as noted, an adaptation to the filtering properties of seawater, but the particular shade also corresponds to the peak sensitivity of the eyes of many deep-sea animals. The photoreceptors in those eyes are most sensitive to exactly the wavelengths that travel farthest through water, and this correspondence is not accidental. Over very long timescales, in a consistently dark environment, the organisms that could detect the faint bioluminescent signals of other organisms and the organisms that could produce signals detectable to potential mates of their own species were the ones that reproduced. The eyes and the lights co-evolved, each shaping the other, until the sensory systems and the signaling systems of the deep ocean arrived at a kind of tuned agreement. Blue-green is the shared channel, the wavelength that matters, the color that means something in the dark. This agreement runs through the ecosystem from top to bottom of the light-producing zones. A small bioluminescent organism at 400 meters produces blue-green light. The predator that eats it has eyes tuned to blue-green. The predator's own photophores produce blue-green light for counter-illumination. The larger predator above it, watching for silhouettes, has eyes oriented upward and tuned to the same range. The whole system, viewed from outside, has the quality of something calibrated, different organisms at different trophic levels, all using the same light, all sensitive to the same wavelengths, all participants in a visual ecology built from the one color the ocean cannot completely absorb before 1,000 meters. In the deepest part of the twilight zone, near 1,000 meters, the photophores of some animals have structures that focus and direct the light they produce, more like a flashlight than a lantern, aimed rather than diffuse. These focused bioluminescent organs appear in several unrelated groups of animals, suggesting that the advantage of directional light, being visible from some angles and not others, concentrating the signal in a specific direction, is significant enough to have evolved more than once. A photophore with a simple reflector is useful. A photophore with a lens and a reflector, capable of directing its light into a narrow beam, is more useful still in an environment where signaling to a specific individual at a specific angle without broadcasting to every predator in the vicinity has survival value. The mesopelagic zone is also home to a large number of animals that produce bioluminescence, not to communicate or to counter-illuminate, but as a defensive response, a last resort when a predator is already close. The ostracod, a small crustacean related to shrimp, when seized by a predator, releases a cloud of luminescent material into the water from glands near its mouth. The cloud lingers, glowing, while the ostracod attempts to escape. The predator, now visibly lit up by the cloud, becomes detectable to its own predators. This is sometimes called the burglar alarm effect. The prey uses light not to hide itself, but to make the predator visible, shifting the danger from the prey to the predator in a single, brief chemical event. Other animals flash brightly when touched, not because the flash discourages the predator directly, but because it startles, giving the prey animal a fraction of a second of disorientation in the predator's grip, enough sometimes to escape. Whether this works consistently is difficult to test in the deep ocean, where behavioral observations require technology that keeps changing. What is known is that a very large proportion of mesopelagic and deep water organisms produce bioluminescence, and that the bioluminescence they produce serves different functions in different species, and that the variety of uses counterillumination, luring, communication, alarm, defense suggests that light in the deep ocean is not a single adaptation, but a toolkit, with different species having reached into it at different times and for different purposes. Near the bottom of the mesopelagic zone, at depths approaching 1,000 meters, the diversity of photophore arrangements in fish is notable. Some species carry photophores only along the ventral surface. Some carry them along the flanks in species-specific patterns. Some have photophores on the head, around the eyes, at the tips of appendages. Some produce light of slightly different colors, still within the blue-green range, but shifted toward the green end or the blue end, in ways that may be significant for species recognition, even when the difference is too subtle for human vision to distinguish reliably without instruments. The study of these small differences is ongoing, and new species with new arrangements continue to be described as trawl surveys cover parts of the mesopelagic zone that have been little studied. The light that enters the ocean at the surface and is progressively stripped away by the water column leaves, in its absence, a world that is not dark in a simple or uniform sense. It is dark with exceptions. It is dark in ways that are carved up by the biology of the animals living in it, each one carrying its own private light, or its own tuned darkness, each one a participant in a visual ecology, as structured and as layered as any ecosystem built on sunlight. The ecology of the sunlit surface ocean is visible and familiar, fish and plankton, and birds and light. The ecology of the deep ocean's dark is structured around luciferin and luciferase, and the blue-green wavelengths that survive the descent, and around eyes shaped over millions of years to find those wavelengths in the dark and make something of them. The dragonfish moves through its midnight zone, unhurried. Its red beam extends forward through water that has not seen sunlight in a very long time, water that has been sitting at these depths, cold and still and dark for months at a time without significant disturbance. The small fish and crustaceans in its path are illuminated in red, in a wavelength their eyes do not register, in a light that to them is simply not there. The dragonfish sees them. It moves toward whatever it has decided is worth moving toward. The dark around it carries its own signals, blue-green flashes from other animals passing at other depths, photophore patterns from the flanks of fish it shares this zone with, brief bright pulses from organisms it disturbs as it passes. All of this light is private in its way. All of it is tuned to a specific sender and receiver, or to a specific moment of use, the lure glowing for whatever approaches, the counter-illumination erasing a silhouette from below, the burglar alarm lighting up a predator who got too close. The deep ocean is threaded with lights like this, moving through the dark at every depth below the surface, wavelengths and signals sent between organisms that evolved to send and receive them over hundreds of millions of years, in the dark, carrying on their quiet exchange in water, so cold and so far from the surface that the presence of any light at all is something built entirely from within, passing through the deep without interference, right now, far below. In September of 2004, a camera rig descended alone through the water north of the Ogasawara Islands. It carried no crew. It was baited, and it was rigged to record whatever came to investigate, and it sank to approximately 900 meters before settling into position in the dark. The researchers aboard the vessel at the surface had no way to watch in real time. They would have to wait for the rig to complete its soak and return before they could see what, if anything, had been recorded. What came to the bait, while the camera recorded alone in the dark, was a giant squid, Architeuthis dux, the first of its kind ever documented alive in its natural habitat. The footage, when it reached the surface, showed an animal that had been living at that depth all along, entirely indifferent to the fact that no human being had ever confirmed its existence there before. The eye that appears in that footage is worth considering on its own terms. Giant squid eyes can reach 30 centimeters in diameter. That is roughly the size of a dinner plate, a single eye, oriented in the dark water, taking in whatever light exists at 900 meters depth. Among all living animals, no eye is larger. The reason for that scale, as best as researchers can determine, is that the squid is not trying to see marine snow or the faint glow of small organisms. It is trying to detect something much larger. Sperm whales hunt Architeuthis dux at depth, and a diving sperm whale produces a faint bioluminescent disturbance in the water, other organisms reacting to its passage. Small lights disturbed and reignited in the whale's wake. An eye large enough to gather those faint, scattered signals from a great distance gives the squid something a smaller eye could not: time. Enough time to move before the whale arrives. The eye is not designed for beauty or for panoramic vision of a sunlit reef. It is a detection instrument calibrated for a specific threat in a specific darkness at a specific depth. Giant squid are members of the phylum Mollusca, making them distant relatives of the garden snail, though the gap between those two animals in terms of scale and habitat is considerable. Architeuthis dux reaches lengths of up to 13 meters in females, and its arms are lined with toothed suckers, capable of leaving circular scars on the skin of the sperm whales that prey upon them. For a long time, the species existed primarily as those scars, and as beaks recovered from the stomachs of caught whales, and as partial carcasses washed ashore. The living animal in its living place was entirely a matter of inference. The 2004 footage, shot by Tsunemi Kubodera and Kyoichi Mori, resolved something that had been genuinely open, not a myth, not an exaggeration, but a real animal whose daily existence had simply been inaccessible to human observation. It continued existing at 900 meters, whether anyone was watching or not. Somewhat deeper in the ocean, between 600 and 900 meters, lives an animal with a name that has caused some confusion since it was first described. The vampire squid, Vampiroteuthis infernalis, is not a squid. It is also not an octopus. It occupies its own separate order within the class Cephalopoda, a lineage that diverged from both the squids and the octopuses long enough ago that it now holds a distinct place in the classification of life. The name reflects its appearance, a dark, cloak-like web of skin connecting its arms, and eyes that appear red under certain lighting conditions. But its habits are far quieter than the name implies. The vampire squid does not hunt. It does not pursue prey through the water or use speed or force to capture anything. It feeds instead on what drifts down from above. Marine snow is the term researchers use for the continuous, slow fall of organic particles from the ocean's upper layers. Dead organisms shed mucus, fecal pellets, fragments of decomposing matter, all drifting downward through the water column in a constant, fine-grained descent. In the zones where the vampire squid lives, this material arrives slowly and in low concentrations. The animal has evolved two retractile filaments coated in mucus that it extends into the water around it, collecting passing particles and drawing them back to its mouth. It is, in effect, a filter feeder operating in near total darkness, at a depth where most animals would struggle to find enough oxygen to sustain themselves. The oxygen minimum zone, where dissolved oxygen in the water reaches its lowest concentration, is exactly where the vampire squid resides, a zone most predators avoid, which may be part of why the vampire squid survives there with such a low-effort feeding strategy. The predators that might otherwise take it cannot comfortably operate at that depth. The vampire squid's metabolic rate is among the lowest of any cephalopod. Its body temperature matches the surrounding water, which hovers a few degrees above freezing at those depths. Its movements are slow and deliberate, with none of the rapid color changes and quick escapes of its shallower water relatives. When threatened, it does not release ink. Instead, it can invert its web of skin back over itself, exposing soft, light-producing organs on the surface of its arms, a display that researchers believe functions as a deterrent. The name Vampirotuthis infernalis translates roughly to the vampire squid from hell, which is a more dramatic label than the animal's actual habits seem to warrant. What it does in the low oxygen dark between 600 and 900 meters is extend its filaments into drifting particles and wait. It is very good at waiting. Not far from the depth range where the vampire squid lives, though scattered across more ocean than it occupies, moves an animal that looks at first encounter as though someone has placed a small pair of green telescopes inside a transparent water balloon and attached a fish to the underside. The barrel-eye fish Macropinna microstoma carries its eyes inside a fluid-filled, transparent dome that encases the top of its head. The dome is not decorative. It serves a structural and optical function, protecting the tubular eyes within and providing a clear medium through which they can gather light. For many years, researchers who studied barrel-eye fish and trawl nets believed the eyes pointed permanently upward, fixed in their sockets in a rigid orientation toward the surface. This seemed reasonable enough. The fish appeared to be scanning the water above it for the silhouettes of small prey against the faint downwelling light. In 2009, researchers at the Monterey Bay Aquarium Research Institute observed living bearlie fish underwater in their natural conditions using remotely operated vehicles. What they found was that the eyes were not fixed at all. The tubular eyes could rotate within the transparent dome, swiveling from their upward pointing position to a forward-facing one when the fish turned towards something ahead of it. This changed the picture considerably. A fish with permanently upward eyes can only see what passes above it. A fish whose eyes can shift to face forward can track and approach prey directly, using the same eyes that scan the water column above to look straight ahead when it needs to. The transparent dome makes this rotation possible by giving the eyes a medium to move through without being obstructed by bony structures that would otherwise constrain them. The fish was doing something more flexible and more precise than anyone had realized, and it had been doing it all along, unobserved, at several hundred meters depth. Macropinna microstoma is small. Adults are typically around 15 centimeters in length. The eyes themselves are large relative to the body, and their distinctive green color comes from a yellow pigment that filters certain wavelengths of light. At the depths where the barrel eye lives, between roughly 400 and 800 meters, the water is dim but not entirely dark. There is still enough faint, diffuse light filtering down from above that a creature with sensitive forward-facing eyes can make some use of it. The fish hovers with minimal movement, oriented in the water column, occasionally rising toward the bells of siphonophores, the long colonial animals that drift through the same depth range to pick off the small crustaceans that gather around them. The transparent dome protects the eyes from the nematocysts, the stinging cells of the siphonophore's tentacles. Even the dome, it seems, has more than one function. The siphonophore requires a moment of separate attention. Priodubia is a colonial organism composed not of a single body, but of hundreds, sometimes thousands, of individual units called zooids. Each zooid is genetically identical to all the others in the colony, but each performs a single specialized function. Some propel the colony through the water, some are responsible for reproduction, some capture and digest food, some produce bioluminescent light. No individual zooid is capable of surviving on its own. The colony moves and feeds and reproduces as a single coordinated structure, and yet it is technically not a single animal in the way that a fish or a squid is a single animal. It is a collective of specialized parts, each one dependent on all the others, operating together with a coherence that resembles from a distance the behavior of one creature. Some specimens of Priodubia have been estimated to exceed 40 meters in total length. This would make them among the longest animals ever recorded, longer than the blue whale, longer than any fish, longer than the giant squid. The structure floats through the water in a long trailing filament, nearly invisible in the dim water of the mesopelagic and bathypelagic zones, its trailing tentacles extending outward into the surrounding water to capture small organisms. The tentacles are not passive. They contain nematocysts that fire on contact, paralyzing small fish and crustaceans, which are then drawn into the colony's digestive zooids. The whole structure operates without a brain, without a central nervous system in the conventional sense, without a single point of coordination. The colony functions because each zooid does its part, and because the parts were shaped by evolution to work together in exactly this arrangement. What makes Priodubia difficult to observe, and therefore difficult to measure accurately, is that the animal is extraordinarily fragile. Trawl nets destroy it. Even the turbulence created by a research submersible can break a long colony into pieces. For most of the history of deep-sea research, siphonophores were known primarily from fragments, partial colonies pulled apart by collection equipment, their true lengths impossible to reconstruct from the pieces recovered. The introduction of remotely operated vehicles with cameras capable of hovering at a distance without generating strong currents changed what researchers were able to see. Long, intact colonies became observable. The estimates of their maximum lengths shifted upward significantly as a result. There may be colonies longer than any yet observed, drifting through the deep water in places where no camera has yet arrived, extending their filaments into the dark in the unhurried way of an organism that has been operating by this method for a very long time. There is a particular quality to the way colonial organisms like Priodubia exist that is worth settling into for a moment. The colony does not have a clear boundary between self and non-self in the way most animals do. A fragment broken from the main body may continue living briefly on its own, performing its one specialized function before failing without the support of the rest of the colony. Whether the colony experiences anything in the way of sensation is genuinely unknown and genuinely unknowable at present. What can be said is that it moves, it feeds, it reproduces, and it has done so continuously for millions of years. in the open water of the deep ocean, in a form that has no real analog anywhere on land. The nearest comparison most people reach for, a long, thin, gelatinous creature drifting in open water, doesn't quite capture what Priadubia actually is, which is something closer to a cooperative society, organized around nothing but function, sustained by nothing but the fact that the arrangement works. The ocean floor at the greater depths hosts an entirely different quality of life, slower, more patient, built for waiting rather than for moving. On the deep sediment below 500 meters and often much deeper lives the giant isopod, Bathynomus giganteus, an animal that bears a striking resemblance to a large armored pillbug, which is precisely what it is in the taxonomic sense. Isopods are crustaceans related to crabs and shrimp, and the terrestrial pillbugs that curl into balls under damp rocks in gardens worldwide are among the giant isopods' closest living relatives. The scale difference is considerable. Where a pillbug reaches a few millimeters in length, Bathynomus giganteus can reach 50 centimeters, roughly the length of a human forearm. The size difference is thought to reflect a phenomenon known as deep-sea gigantism, in which reduced predation pressure, cold temperatures, and the metabolic effects of high-pressure environments allow certain crustaceans to grow to much larger sizes than their shallow water counterparts. The giant isopod is not a fast animal. It moves across the seafloor in a slow, deliberate walk, its 7 pairs of legs carrying its segmented body over the sediment in a manner that conveys no urgency at all. It is an opportunistic scavenger, feeding on the carcasses of whales, fish, and squid that settle to the seafloor, as well as on whatever other organic material arrives. In the deep ocean, the arrival of a large food source, a whale fall for instance, is an irregular event, unpredictable, and widely spaced in time. The isopod's entire physiology is built around this irregularity. It can eat an enormous amount when food is available, and it can go for extraordinary periods without eating at all. One captive specimen held at Toba Aquarium in Japan went without food for 5 years and 2 months before it died, which was notable not because it starved over that time, but because it simply did not eat. It remained alive throughout. Whether it was waiting in any sense that word implies cannot be known with certainty. The 5-year fast is the kind of fact that takes a moment to settle into place. Most familiar animals, cats, dogs, birds, even most fish, require food on a scale of days to weeks. An animal capable of sustaining itself for 5 years without a single meal is operating under a set of metabolic rules that don't correspond to anything in ordinary experience. The isopod in the Japanese aquarium was observed and healthy throughout most of that period. It moved occasionally. Its vital signs appeared normal. It simply did not eat. The mechanism is not fully understood. Researchers believe the isopod is capable of drastically reducing its metabolic rate during periods of food absence, drawing on fat reserves stored from previous meals. But the full physiology of extended fasting at this scale has not been completely mapped. What is clear is that the animal is built for a world where food is sparse, where meals may be years apart, and where patience is not a choice, but a structural feature of the organism itself. Bathynomus giganteus has compound eyes, like those of most crustaceans, large, fixed, reflective eyes, positioned on either side of the head, designed to detect light and movement rather than to form detailed images. At the depths where the giant isopod lives, there is very little light of any kind, only the occasional faint glow of a bioluminescent organism passing overhead, or the chemical trail of an organic particle drifting down through the water. The eyes may function primarily to detect that bioluminescence, alerting the animal to the presence of other organisms nearby. Beyond the eyes, the isopod has two pairs of antennae that it uses to sense chemical signals in the water: the smell of a whale carcass, the trace of decaying tissue carried on whatever current exists at depth. The antennae are the more important sensory organs in a world defined by darkness, and the isopod keeps them in near constant slow motion, reading the water for information about what might be nearby. The segmented body of Bathynomus giganteus is protected by a series of overlapping plates, flexible at their joints but rigid in themselves. The same basic plan as the terrestrial pillbug, scaled up and hardened for a different set of pressures. The isopod can roll itself into a ball, curling its body so that the armored plates face outward in all directions, a defensive posture that works as well at 500 meters depth as it does in a garden. Whether it uses this posture much at depth, where predation pressure is lower than in shallower water, is uncertain. The structural capacity is there regardless. The animal carries the defensive architecture of its shallow water ancestors into an environment where it may rarely need it, in the way that many deep-sea organisms carry adaptations shaped by the environments of earlier epochs, before the lineage descended into deeper water. There are other large crustaceans in the deep ocean: spider crabs, certain shrimp reaching considerable size, king crabs in some regions, but none combine the scale, the solitary patience, and the observable stillness of Bathynomus giganteus in quite the same way. Most deep-sea crustaceans are active scavengers or active predators, moving through the water or across the sediment in search of the next meal. The giant isopod can do this too, and does when food is available, but it can also simply stop. It settles onto the sediment, tucks its legs slightly, and remains motionless for hours or days or longer. The stillness is not the stillness of an animal that has died or that is impaired. It is the stillness of an organism doing exactly what it has evolved to do in an environment where movement consumes energy. Energy is limited, and the best strategy is often to wait. Returning upward in the water column, to the depths where the vampire squid hovers, and the barrel eye scans the water above it, the ocean between 400 and 1,000 meters has a quality of diffuse, unstructured space that is hard to describe in terms of anything familiar. There is no floor to stand on at those depths, or there is, but it is hundreds of meters further down. There is no surface overhead in any useful visual sense. The light from above is a dim, directionless glow rather than a visible sky. The animals that live at these depths move through open water in all three dimensions, and many of them spend their lives without ever touching either the seafloor or the surface. They exist in a column of water, held in place by their buoyancy and by the behavioral patterns that keep them at their preferred depths, and the world around them is water in every direction, extending for thousands of kilometers horizontally and hundreds of meters vertically, bounded above by light and below by darkness and cold. In this open, unstructured space, the giant squid is not actually the largest active hunter. Sperm whales, which descend regularly to 1,000 meters and occasionally to depths beyond 2,000 meters, move through the same water in search of exactly the kind of prey that Architeuthis dux represents. The relationship between sperm whale and giant squid is among the more thoroughly documented predator-prey interactions in the deep ocean, primarily because the whales carry evidence of it to the surface: circular scars from squid suckers on the skin around their heads, squid beaks accumulated in their stomachs, the chemical traces of squid consumption in the waxy substance of their intestines. The squid itself leaves less evidence. Its body is soft, perishable, and rarely survives long enough after death to wash ashore intact. What is known about its life comes primarily from fragments, beaks, arms, isolated tissues, and from that single sequence of footage captured at 900 meters in 2004. The relationship between the squid's enormous eye and the whale's bioluminescent wake is one of the more elegant adaptations in the deep ocean, in the sense that it involves one animal evolving a sensory organ specifically tuned to detect the presence of another. The sperm whale does not produce bioluminescence itself. It is a large, warm-blooded mammal with no light-producing cells, but its passage through the water disturbs the small bioluminescent organisms around it, causing them to flash. An eye of 30 centimeters diameter, with the optical sensitivity implied by that scale, can detect that disturbance at a distance. The squid does not need to wait until the whale is close. It has time to move if its eye is good enough. The evolutionary pressure that produced those enormous eyes was the whale, not the whale as an abstraction, but the specific recurring pressure of a large predator descending from above, in a darkness where vision is the only warning. The barrel eye and the vampire squid, and the giant squid, and the siphonophore all occupy different niches in the same general depth range, overlapping in altitude within the water column, sharing some of the same dark, cold, dim to lightless space, but each occupying it in a different way and eating different things by different means. The barrel eye picks crustaceans from the periphery of siphonophore colonies. The vampire squid filters organic particles from the water with its mucus-coated filaments. The siphonophore captures small fish and crustaceans with trailing nematocyst-bearing tentacles. The giant squid pursues prey large enough that it requires the kind of muscular arms and toothed suckers that have left their marks on sperm whale skin. The same dark, cold, oxygen-variable water column supports all of them, and they go about their different businesses largely without interfering with one another. The niches are separate enough that competition between them is limited. What connects all five of these animals, the giant squid, the vampire squid, the barrel eye, the siphonophore, the giant isopod, is not a shared appearance or a shared depth or a shared behavior. What connects them is the environment that shaped them: cold, dark, pressurized, food sparse, and slow. Each one carries adaptations that are incomprehensible without that environment as context. The enormous eye that makes sense only in near total darkness. The low oxygen tolerance that makes sense only in the oxygen minimum zone. The transparent dome that makes sense only in dim water where rotating eyes can still gather usable light. The colonial structure that makes sense only in open water where no single body plan could be both propulsive and reproductive and defensive all at once. The 5-year fast that makes sense only in a place where the next meal might be years away. The giant isopod at rest on the sediment is in its stillness a reasonable image to rest with. The segmented body is low and flat against the floor, the antennae moving in slow arcs through the water, the compound eyes absorbing whatever faint information the surrounding dark offers. It is not waiting for anything in particular. It is not expecting anything. It is simply present in the environment it was built for, doing what it has always done at a depth that most organisms cannot survive, in a darkness that most organisms have never encountered. The legs are folded slightly beneath the body, not fully retracted, in the posture of an animal that could move if it needed to, but currently does not need to. The sediment around it is undisturbed. The water is very still and very cold. There is no sound, or there is sound. the slow, low creaking of pressure that humans cannot hear, but that may register somewhere in the sensory apparatus of a crustacean evolved for these depths. The five animals in this section are not the only extraordinary inhabitants of the deep open ocean. They are five among thousands of described species, and thousands more that have not yet been described. They were chosen because each one opens a slightly different window onto what life in cold, dark, pressurized water actually looks like, not as an abstract category, but as a set of specific, documented, observable facts. The squid with the dinner plate eye, watching for the bioluminescent wake of a whale. The vampire squid extending its mucus-coated filaments into a slow drift of organic particles. The barrel-eye rotating its tubular eyes inside a transparent dome as it turns towards something ahead. The siphonophore trailing 40 meters of cooperative zooids through open water. The isopod sitting still on the sediment, its metabolic rate dialed down to something close to minimum, fully capable of remaining in that posture for months or years before the water carries the scent of something worth moving toward. These are not exceptional animals in the sense of being rare or difficult to find, though many of them are genuinely difficult to observe. They are exceptional in the sense that they exist at all, in the specific forms they have taken, shaped by pressures and constraints that have no equivalent anywhere on land. The cold did something to them. The dark did something to them. The pressure did something to them. The irregular, unpredictable, slow arrival of food did something to them. What resulted across millions of years was this: a set of living things that carry the deep ocean inside them in the specific shapes of their eyes and bodies and metabolic strategies, perfectly suited to a world that most of the rest of life on Earth will never enter. The isopod does not move. The antennae continue their slow arcs. The water above it is cold and still, extending upward through hundreds of meters of dark before the first faint traces of light begin to appear from the surface. In the water column above the isopod, somewhere between 600 and 900 meters, a vampire squid extends its filaments and draws them back slowly. Further up, in the dimming twilight zone, a barrel-eye fish holds its position in the water, its green tubular eyes pointing upward through the transparent dome of its head, waiting for the silhouette of something small to pass between it and the faint filtered light descending from above. These animals are there right now. The darkness they inhabit is not interrupted by this observation. The squid does not know it was filmed in 2004. The isopod in a Japanese aquarium that went 5 years without eating did not know it was being recorded. They are exactly what they are, doing exactly what they do, in the specific cold and dark and pressure they were made for, entirely indifferent to the fact of being remarkable. The ocean holds them in place, and they hold their positions, and the water above them moves slowly, and the sediment below accumulates, layer by quiet layer, exactly as it has for longer than the names we have given these animals. A single parcel of water somewhere in the middle of the North Atlantic is doing something that takes 1,000 years to finish. It has traveled up from the tropics along the ocean's surface, carried north by wind and the slow curve of the Earth's rotation, and now it is arriving in the cold air above the Labrador Sea in winter, losing its warmth degree by degree to the atmosphere above it. The water grows dense. It grows heavy with salt, not because anything has been added to it, but because so much heat has left it that its molecules pack more closely together, and the salt that was always there now sits in a slightly smaller volume of water. And then, without any particular event marking the moment, it begins to sink. It does not sink quickly. It descends at a rate that would be invisible to any observer, a fraction of a meter per day, in some stretches, slower still in others. but it descends without stopping, and it will go on descending until it reaches the deep ocean floor, and then it will spread south and east across the Atlantic basin, around the tip of Africa, eastward through the Indian Ocean, northward and southward through the Pacific, in a route that will take between 500 and 2,000 years to complete. Oceanographers call this the thermohaline circulation. The name comes from the two forces that drive it, thermo for heat, haline for salt. But names aside, what the circulation amounts to is the ocean moving itself through itself, trading deep water for surface water and surface water for deep, carrying heat from the equator toward the poles and cold from the poles toward the equator, in a cycle that has been running without interruption since the last time the ocean's chemistry changed substantially enough to alter it. The water moving through this system is not marked. There is no dye in it, no flag attached to any particular parcel, as it makes its 1,000-year circuit. What researchers know about the circulation comes from measurements of temperature, salinity, and dissolved oxygen, properties that change predictably as water ages and travels, and that allow oceanographers to distinguish a water mass that formed in the North Atlantic last century from one that formed near Antarctica several hundred years before that. The circulation has been reconstructed from those measurements, slowly and over many decades, a picture assembled from countless individual data points taken from research ships and moored instruments spread across every ocean basin. The two places where deep water forms most reliably are the North Atlantic and the waters around Antarctica. In the North Atlantic, the process is essentially the same one described above: warm surface water traveling north, losing its heat, growing dense, sinking. The water mass that results from this process has a name: North Atlantic deep water. It forms near Greenland, near Iceland, and in the Labrador Sea, and it sinks to depths between 2 and 4 kilometers before spreading south and east through the deep Atlantic. In the Southern Ocean, around the edges of Antarctica, a slightly different version of the same process occurs. When seawater freezes into sea ice, the salt it contained is largely expelled back into the surrounding ocean, making the remaining liquid water saltier and denser still. That cold, salt-heavy water sinks along the Antarctic continental slope and spreads northward along the ocean floor into the Pacific, Atlantic, and Indian basins, filling the very deepest parts of each. This water mass is called Antarctic bottom water, and it is, by most measures, the densest water in the world's oceans. Both of these water masses formed at the surface. Both of them carried dissolved oxygen down with them when they sank. One of the ways researchers can trace how old a body of deep water is, how long since it was last at the surface, is to measure how much of that original oxygen remains. Deep water breathes. In a sense, the organisms living in it consume oxygen over time, and the longer a water mass has been isolated from the surface, the less oxygen it contains. A body of water in the deep Pacific that has not seen the surface in 1,000 years will carry the chemical signature of that long isolation, a low oxygen reading that tells oceanographers something about where it formed, how long it has been traveling, and roughly when it might return. The pace of the circulation matters in ways that extend far beyond the ocean itself. One of those ways involves heat. The deep ocean is cold. Temperatures in the abyssopelagic zone sit near 2 degrees Celsius, in some places lower, and the process of forming deep water essentially sends warm surface water down into that cold reservoir, replacing it with upwelled cold water at the surface. That exchange is part of what keeps the tropics from warming indefinitely, and what delivers heat to regions far from the equator that would otherwise be considerably colder. Northern Europe, whose latitude places it far poleward of where its climate would otherwise suggest, receives warmth, in part because the Atlantic circulation carries heat northward along the surface before the water sinks. The relationship is not simple, and the ocean is not the only factor, but the circulation is a piece of the planetary heat budget that runs at a scale and on a time scale that dwarfs any individual weather event. The deep ocean has another role in the planetary system, and it is one that operates through chemistry rather than heat. The ocean absorbs carbon dioxide from the atmosphere at its surface, a process driven partly by simple physics and partly by the activity of tiny photosynthetic organisms near the surface that take carbon out of the water to build their bodies. When those organisms die, some fraction of them sinks. Their carbon-containing remains drift downward through the water column, gradually, through hundreds or thousands of meters, joining the continuous slow fall of organic matter from the sunlit surface. This material, fragments of dead organisms, fecal pellets, mucus aggregates, all of it bound loosely together and drifting, settles toward the seafloor, and some fraction of the carbon it contains is incorporated into the sediment when it arrives. The carbon locked into that sediment is effectively removed from the atmosphere for geologically significant periods of time, thousands to millions of years depending on whether and how the sediment is eventually subducted or uplifted. Oceanographers and biogeochemists call this the biological carbon pump. The pump is not a machine in any conventional sense. It is the accumulated effect of an enormous number of small events: an organism dying, a particle sinking, a molecule of carbon settling into mud, each one trivial, and each one part of a process that moves carbon out of the atmosphere and into the seafloor at rates that have shaped the composition of Earth's air over geological time. The deep ocean has been running this process continuously for hundreds of millions of years. The sediment layers accumulating on the abyssal plain are, in a sense, a record of it. Each thin layer representing decades or centuries of organic material drifting down and arriving, compressed slowly into something solid enough that a research core can pull it up and read it, the way tree rings are read, as a record of conditions at the surface of the ocean, long before any instrument was there to measure them. The abyssal plain itself is worth pausing on. It lies between roughly 3,000 and 6,000 meters depth, and it covers something close to 50% of the total surface area of the Earth, more than all the continents combined. It is by most reckonings the largest single type of habitat on the planet, and most of it has never been directly observed. From above, it receives that slow, continuous fall of organic particles from the surface 2 or more kilometers higher. From below, it rests on oceanic crust that varies in age from quite young, near the mid-ocean ridges where new crust is constantly being created, to quite old, where it has been moving away from those ridges for tens of millions of years. The sediment that has accumulated on that crust varies in thickness accordingly: thin near the ridges, deep and old near the continental margins, where the drift of fine particles has had the longest time to accumulate. The floor of the abyssal plain is not featureless. There are valleys and low swells, ancient impact craters filled with sediment, seamounts rising hundreds of meters from the surrounding floor, and the traces of submarine landslides on the slopes of underwater ridges. But the dominant character of the place is flatness, a flatness more complete than almost anywhere on the continental surface, because the processes that roughen land over time, erosion and tectonics, and the movement of ice operate differently, or not at all, at 4,000 meters in the dark. What shapes the abyssal plain shapes it slowly: the accumulation of sediment, the occasional disturbance of a submarine current, the passage of a turbidity current down a canyon that pushes a fan of material outward across the floor. Between those events, the plane simply waits, in the way that flat and quiet places wait, accumulating its thin layers of arriving carbon, holding the temperature of the water above it at just above freezing. The rain of organic matter reaching the abyssal plane is light by any surface standard. In the most productive parts of the ocean, above upwelling zones where cold, nutrient-rich water rises to the surface, the flux of material reaching the deep floor is measurably higher than in the open gyres, where the surface is warm and nutrient-poor, and the biological productivity is relatively low. In the open subtropical gyres, which cover enormous areas of the Pacific and Atlantic, the abyssal floor receives so little organic material that the sediment accumulates at rates of a few millimeters per 1,000 years. A layer of sediment 1 centimeter thick in those regions may represent 10,000 years of slow, steady arrival from the world above. Sitting inside that sediment, in those thin annual layers, are the shells and tests of countless microscopic organisms, foraminifera, coccolithophores, radiolarians, each one a tiny record of the temperature and chemistry of the surface water it formed in, preserved in calcium carbonate or silica, still intact after millions of years of burial. When researchers pull a sediment core from the abyssal plain, they are recovering a continuous record of the ocean's surface conditions stretching back, in some cases, tens of millions of years. The deep ocean floor is one of the planet's best archives. It holds its records quietly, in the dark, accumulating new pages at a rate too slow to observe in any human lifetime, pressing the past steadily downward as the present settles onto it from above. The cold that defines the deep ocean does not stay still. It moves, though the movement is too slow and too diffuse to experience directly. Antarctic bottom water, once it has formed and sunk along the continental slope, does not simply pool in place. It spreads northward along the floor of every deep basin, pushed by the density contrast between it and the slightly less dense water above it, flowing along the bottom the way cold air flows along a floor. In some basins, oceanographers can trace this flow as a measurable current close to the seafloor, a bottom water current carrying the chemical signature of its Antarctic origin, very cold, very salty, very low in oxygen-consuming age, far into the northern hemisphere. The deep basins of the ocean are not open containers. They are divided by underwater ridges and sills, elevated sections of the seafloor that act as barriers to the densest bottom water. Water that is dense enough to overflow a sill will do so. Water that is not will pool on the colder side of it, unable to continue. The result is that different deep basins contain water of different ages and different temperatures, depending on how tall their bounding ridges are and how freely bottom water can flow across them. The deepest parts of some isolated basins contain water that has been cut off from the general circulation for a very long time, sitting in place as the ocean above it slowly exchanges its upper layers, aging quietly at the bottom of a natural enclosure. The heat the ocean absorbs does not stay at the surface. Over time, some of it penetrates downward, warming the upper layers of the ocean at rates that can be measured by instruments held at fixed depths over years and decades. The deep ocean has absorbed more than 90% of the excess heat that has accumulated in the Earth's climate system over the past several decades, acting as an enormous thermal reservoir whose capacity dwarfs anything at the surface. The ocean's heat capacity is large enough that it can absorb heat steadily for years without the surface temperature rising as quickly as it otherwise would, a thermal inertia that affects the pace of change at the surface. distributing the effect of that added heat across the entire volume of the ocean, which is large enough that even significant additions of energy produce relatively slow distributed warming. The way the ocean stores heat is somewhat like the way a thick stone wall stores the warmth of an afternoon. Sunlight falls on the stone through the morning and afternoon. The surface warms, and slowly the heat works its way inward, deeper and deeper through the stone, so that hours after the sun has set, the interior of the wall is still releasing warmth it absorbed earlier. The ocean does something similar at a vastly larger scale, absorbing heat at the surface over years and decades, distributing it through the water column slowly, releasing it in places and at times distant from where and when it arrived. The Southern Ocean, Minukstutation, the Southern Tsomien, Dutation Southernware, Medelofgersugrisheni, the Tiritzlodsritbething which circles Antarctica without interruption from any continental landmass, is one of the most active regions of this heat uptake. The deep cold water upwelling there creates a steep thermal gradient that drives absorption of both heat and carbon dioxide at rates per unit area higher than most other ocean regions. The forces driving water through the deep ocean are not dramatic to observe. There is no moment at which the thermohaline circulation becomes visible from the surface, no current you could identify by color or turbulence or feel. The movement of deep water is revealed not by anything it looks like, but by what it contains, the temperatures and salinities and dissolved gases that oceanographers measure and map and trace as indirect markers of motion. In this way, the deep ocean circulation is somewhat like watching traffic by tracking registration plates across a continent, rather than by seeing any individual car move. What appears when enough data is assembled is a coherent pattern, a global circulation with identifiable formation zones, transport pathways, and upwelling regions where deep water eventually returns to the surface and begins the cycle again. That upwelling is the closing part of the circulation. Deep water that sank in the North Atlantic hundreds or thousands of years ago eventually rises back toward the surface, largely in the Southern Ocean and in the equatorial Pacific and Indian Oceans, where the geometry of the seafloor and the patterns of wind-driven surface currents create the conditions under which deep water is drawn upward. When it arrives at the surface, it is cold, dense with the accumulated nutrients of centuries at depth, and relatively poor in oxygen compared to newly formed deep water. It spreads across the surface, warms, absorbs oxygen from the atmosphere, takes up carbon dioxide, and eventually, somewhere poleward, begins to cool and sink again. The mid-ocean ridges play a different role in the planet's deep ocean story, one that operates at a longer timescale still, measured not in centuries, but in tens of millions of years. A mid-ocean ridge is a divergent boundary between tectonic plates, a place where two sections of oceanic crust are moving apart, and where new crust is being continuously pressed upward from below to fill the widening gap. The material that fills that gap is magma from the mantle, which cools and solidifies as it contacts the surrounding seawater, adding new oceanic crust to the edges of the separating plates, and pushing the older crust outward in both directions. The mid-ocean ridges form the longest mountain chains on Earth. Together they run approximately 65,000 kilometers through every ocean basin on the planet, through the Atlantic from north to south, through the Indian Ocean, through the Southern Ocean around Antarctica, and through the Pacific where the ridge system runs closer to the eastern margin than the center. In the Atlantic, the ridge is roughly centered between the continents, a long sinuous elevation rising 2 kilometers above the surrounding abyssal plain, its crest marked in many places by a central rift valley where the separation between plates is actively occurring. In the Pacific, the ridge is broader and lower in profile, spreading faster and producing crust at a higher rate. None of this is visible from the ocean surface. The peaks of the mid-Atlantic ridge reach several kilometers below the surface at their highest, nowhere breaking into open air. Ships pass over the ridge routinely, their keels skimming the surface of an ocean whose floor is 2.5 kilometers below, a mountain chain the size of which has no equivalent on land. The Rocky Mountains, the Andes, the Himalaya, all are shorter in total length than the mid-ocean ridge system by an order of magnitude. The ridge exists in the dark, on the floor of the deep ocean, building itself outward in both directions at a pace measured in centimeters per year. The seafloor created at the ridges is young when it forms and grows older as it spreads away. The oldest oceanic crust on Earth is around 200 million years old, found in the western Pacific in regions where the floor has been moving away from its creation point since the Triassic. Continental crust, by comparison, can be 4 billion years old or older. Oceanic crust is geologically short-lived because it is eventually subducted at convergent boundaries, pulled back into the mantle at the deep ocean trenches, recycled into the same material it came from. The ocean floor is continuously being remade, new crust at the ridges, old crust disappearing at the trenches, and the sediment that accumulated on top of it over millions of years goes down with it, returning the carbon locked into those sediment layers to the deep interior of the Earth. The water in the deep ocean is affected by the ridges in ways beyond simple geography. Hydrothermal circulation drives seawater down through cracks in the oceanic crust near the ridges, where it is heated by magma below, altered chemically by reaction with hot rock, and returned to the ocean through vents. This process occurs continuously along the entire length of the ridge system, and the chemical exchange it drives, magnesium and sulfate leaving the water, calcium and dissolved metals entering it, affects the chemistry of the entire ocean over long timescales. The ridges are not merely the place where the ocean floor is made. They are an active chemical interface between the ocean and the solid earth beneath it, running without pause at the bottom of an ocean that covers most of the planet's surface. The deep ocean's slow work extends into the question of climate over geological time in ways that research is still assembling. The biological carbon pump, over millions of years, has moved carbon from the atmosphere into the seafloor, and the rate at which it has done so has varied with ocean circulation, surface productivity, and the chemistry of the water itself. Periods of ocean stagnation, when deep water formation weakened and the circulation slowed, have been associated with reduced oxygen in the deep ocean and altered patterns of carbon burial. The sediment record from the abyssal plain carries the signatures of those events in layers of different composition, color, and organic content, readable to geochemists who know what they are looking for. The patience required to read that record is itself worth noting. A researcher working with a sediment core from the deep abyssal plain might be examining material that accumulated over 10 million years, a column of mud perhaps 50 or 60 meters long, representing 10 million years of slow arrival from the surface. Each centimeter of it covers in some regions 10,000 years. The careful work of identifying what organism produced each microscopic shell, measuring the isotopic ratios locked in that shell's calcium carbonate, and placing that measurement in a timeline reconstructed from dozens of other cores. That work takes months or years per core, and the picture that emerges is assembled from thousands of cores collected over decades by researchers on hundreds of ships, making measurements in every ocean basin on Earth. The deep ocean holds that record without any concern for whether it will be read. The sediment accumulates. The cold water sits above it in the dark. The carbon locked into the floor stays locked, in the ordinary course of things, until the floor itself is subducted, or until some disturbance at the surface sends a turbidity current rolling down a submarine canyon to disturb the arrangement. In the meantime, the layers press down on what is below them, the newest material arriving quietly from above, the oldest material growing slowly into rock at the base of the column. What the thermohaline circulation moves through the deep ocean, alongside heat and dissolved gases and nutrients, is time, a water molecule that sank in the North Atlantic in the 15th century, is somewhere in the deep Pacific now, in a water mass whose other molecules have similar histories, formed near Greenland or near Antarctica, sunk in the cold, traveled slowly south and east and north through the deep basins, carrying the dissolved signature of the surface world they left behind. When that water eventually upwells back to the surface, perhaps in the Southern Ocean or in the equatorial Pacific, it will have been in the deep ocean for somewhere between 500 and 2,000 years. It will arrive at the surface, having last seen sunlight during a period when much of the world's population had no idea the ocean circulated at all. The carbon the deep ocean has removed from the atmosphere over geological time amounts to a quantity that is hard to make felt without numbers, and the numbers themselves are large enough to be difficult to hold. Oceanographers estimate that the ocean contains roughly 50 times as much carbon as the atmosphere, most of it dissolved in the water column as bicarbonate and carbonate ions. The sediment on the ocean floor contains more still, calcium carbonate shells accumulated over hundreds of millions of years, compressed into limestone and chalk in many places, a geological record of the biological carbon pump running without interruption across deep time. The deep ocean's role in regulating Earth's climate is not a recent discovery, but the details of how it works and how sensitive it is to changes at the surface have been an active area of oceanographic research for decades, and the picture continues to develop. What is established is that the deep ocean is not passive. It does not simply receive whatever the surface sends down. It participates in the planet's chemistry, circulates its water and heat and dissolved gases on timescales that span human history many times over, and maintains a physical and chemical state that has been shaped by hundreds of millions of years of geological, biological, and atmospheric history. It is the largest continuous feature of the planet's surface, the largest reservoir of liquid water, the largest habitat by volume, and the largest absorber of heat in the current climate. And most of it, the cold, dark, slow-moving interior, remains effectively unobserved, known through measurement and inference rather than direct sight. The abyssal plain sits in the dark, receiving its slow rain of particles from the surface, its temperature sitting just above freezing, its water moving in currents too slow to feel. The ridges press new crust outward in both directions. The Antarctic bottom water spreads northward along the floor of every deep basin, filling the cold bottom of the ocean from the south. The biological carbon pump moves its small particles downward, continuously, each one carrying a fraction of the carbon the surface ocean drew from the air. The thermohaline circulation moves its vast parcels of water through the deep basins, trading heat between ocean and atmosphere, keeping the polar regions warmer than they would otherwise be, and the tropics somewhat cooler. All of this is happening now, while the surface of the ocean sits quiet under whatever weather is passing over it. A ship crossing the North Atlantic leaves a wake that disappears in minutes, and the ocean surface closes over it as though the ship had never passed. Below, at 3,000 meters, the same water that will one day carry the signal of a surface event, a change in temperature, a change in salinity, has not yet felt anything from above. It sits in the dark at close to freezing, moving southward along the floor of the Atlantic at a pace that will bring it to the equator sometime in the next several hundred years. The mid-ocean ridge below it is pressing new crust outward in both directions, a centimeter or two per year, as it has been doing for tens of millions of years. The ridge rises 2 kilometers above the abyssal plain. To either side of it, its central rift valley sitting at the exact line where one plate ends and another begins, new rock cooling and solidifying as it arrives from below and is pushed sideways to make room for what will come next. The mountain chain is 65,000 kilometers long and almost entirely unmapped at high resolution, its peaks and valleys known in outline from sonar sweeps, but unvisited by any camera in the vast majority of their length. The ridges run in quiet and in darkness, the longest mountain range on the planet, building the floor of the ocean from below. The ocean does not require observation to continue its work. The thermohelian circulation does not pause in the absence of measurement. The biological carbon pump moves its particles, whether or not any instrument is counting them. The Antarctic bottom water spreads northward along basins whose floors have been accumulating sediment for tens of millions of years. And the water above those basins sits in the cold dark at temperatures only a degree or two above freezing, moving with the slowness that characterizes all deep ocean movement. Steady, continuous, patient in the way that only very large and very old systems are patient. There is something quietly settling about the scale of it. The circulation that moves a parcel of North Atlantic water through the deep ocean and back to the surface in 1,000 years has been running in some form since long before the first human saw the ocean surface. The sediment on the abyssal plain that contains the carbon of organisms that lived tens of millions of years ago has been lying on that floor in the dark for all of recorded history and all of human prehistory, and for an enormous length of time before either. The ridges have been pressing new seafloor outward since before the continents were arranged anything like the way they are now. The deep ocean is large and old and slow and largely cold. Its work is planetary and its timescale is geological. It absorbs heat and carbon, it circulates water, it builds seafloor and buries carbon, and regulates the chemistry of the atmosphere through processes so gradual they are invisible to any individual human lifetime. What the deep ocean does on any given day is indistinguishable from what it did 1,000 years ago or 1,000 years from now. It moves, it sinks, it spreads, it accumulates, it presses new rock outward from the ridges and moves old rock toward the trenches. And between the ridges and the trenches, across the enormous flat darkness of the abyssal plain, it holds its record of the surface world in thin layers of settling carbon, patient and undisturbed, in the dark, beneath miles of open water that are themselves mostly quiet, moving in their own slow way, carrying the heat and chemistry of the past forward into a future that the deep ocean is, in its way, already preparing for. The mid-ocean ridges extend through every ocean basin, building the floor of the world in the dark, pressing outward in silence, a centimeter at a time, as they have been doing for longer than the ocean's current shape has existed. The longest mountain range on Earth, entirely beneath the water, still and slow and active all at once, making a planet that is, in the deepest and quietest sense, still quietly making itself. On the morning of January 23rd, 1960, 2 men sat inside a steel sphere roughly the size of a small closet, resting on the floor of the Challenger Deep. The sphere was called the Trieste. Outside the viewport, the sediment was undisturbed. The water was completely still. Jacques Picard and Don Walsh had descended nearly 11 kilometers through the water column over the course of 4.5 hours, and now they sat at the deepest point on the surface of the Earth, and the dark outside their window was total. This pressure against the hull was equivalent to the weight of an ordinary family car pressing down on every square centimeter of steel that enclosed them. They had 20 minutes. Picard looked through the viewport and reported seeing a flatfish resting on the sediment a short distance away. It was small and pale, lying quietly on the floor of the world's deepest place, doing nothing in particular, not alarmed, apparently, by the lights of the Trieste, not disturbed by the arrival of the first humans ever to reach that particular patch of ocean floor. It simply rested there, as it presumably had before the submersible arrived, and would after it left. The fish has been described in various ways in the years since. Its exact species has been a matter of some debate, but the image itself has persisted. A small creature at rest on the sediment at the bottom of the deepest trench on Earth, observed through a porthole by two men who had nowhere further to go. They sat for 20 minutes. Then they began the long ascent back to the surface, which took another 4 hours, and the flatfish remained below, and the Challenger Deep returned to the stillness it had maintained for millions of years before the Trieste arrived, and would maintain for some considerable time after. That dive was the first, and for decades, the last of its kind. No one returned to the full depth of the Challenger Deep in a crewed vessel for more than 50 years. The ocean floor at that depth is not a place that humans have found easy to reach, and the difficulty is not merely technical. It is a matter of the sheer volume of water that lies between the surface and the bottom, and the patience required to cross it, and the brevity of whatever can be accomplished once there. The Trieste carried its 2 passengers to the deepest point ever reached by humans, and the 2 passengers had 20 minutes to look out the window, and then they came back up. What remained below them in those 20 minutes, and in the decades afterward, was very nearly everything. The bathyscaphe Trieste had been built with a specific kind of engineering logic, a large float filled with gasoline, which is less dense than water and provides buoyancy, attached to a heavy steel observation sphere below. It was not a nimble vehicle. It could not hover, could not maneuver with precision, could not collect samples with a mechanical arm. It was, in a practical sense, an elevator, something that could carry 2 people down and back and allow them to observe, briefly, what was at the bottom. That it accomplished even this was the result of years of engineering work, and the descent on January 23rd was not without incident. A plexiglass window cracked on the way down, producing a loud bang in the sphere, and for a moment neither man knew what had broken. It had not been the pressure hull. They continued down. The specific courage required to continue downward after an unexplained bang inside a pressure vessel at several thousand meters of depth is a particular kind of courage, one that does not announce itself or require any dramatic gesture, but simply continues. Don Walsh, who was a Navy lieutenant at the time, and Jacques Picard, who was the son of the Swiss physicist Auguste Picard, who had designed the bathyscaphe, sat with the cracked window and continued the descent. The ocean floor came up to meet them. The flatfish was there. 20 minutes later, they began to rise. In the years following the Trieste's dive, most deep-sea exploration shifted toward a different kind of vehicle. The crewed submersible Alvin was commissioned in 1964, operated by the Woods Hole Oceanographic Institution, and has since completed more than 5,000 dives. It is a more capable vehicle than the Trieste in most practical senses. It can maneuver, collect samples, carry instruments, and remain at depth for hours rather than minutes. Alvin is about 7.5 meters long, carries a pilot and 2 observers, and descends at a rate that places it at 4,000 meters in roughly 2 hours. It has been lost at sea and recovered. It has carried scientists to the wreck of the Titanic, and it has, over its 6 decades of operation, returned more scientific data from the deep ocean than perhaps any other single instrument built for the purpose. In February of 1977, Alvin carried a team of scientists to the Galapagos Rift, a section of the mid-ocean ridge system in the eastern Pacific, at a depth of around 2,500 meters. The scientists expected to find what most of the deep sea floor was understood to contain at the time: cold, quiet, mostly empty water above a sediment plain with relatively sparse life. What the lights of Alvin illuminated instead, as the submersible approached the rift, was a seafloor crowded with life. Giant white tube worms, some more than 2 meters long, clustered around warm water rising from cracks in the volcanic rock. Large white clams lay scattered across the sediment. Pale crabs moved among them. The water coming from the vents was warm, in some cases very warm, and it was laden with hydrogen sulfide and other chemicals, and the organisms gathered around it were not living on sunlight filtered down from the surface. They were living on chemistry. The scientists aboard Alvin that day were looking at an ecosystem that nobody had known existed. The tube worms have no digestive tract. They house bacteria in a specialized organ that converts hydrogen sulfide into the energy needed to sustain the worm's tissue. The clams do something similar. Their gills are packed with chemosynthetic bacteria that do the metabolic work. The clams themselves cannot. The entire community around the hydrothermal vents is built on a biological foundation that has nothing to do with sunlight, and finding it meant that the assumptions underlying most of what was understood about life on Earth had to be reconsidered, at least in part. Life, it turned out, did not require a star. It required chemistry and heat and the slow movement of water through warm rock, and there was apparently plenty of that in places nobody had thought to look. Alvin has been through several upgrades in the decades since the Galapagos Rift discovery. Its titanium personnel sphere was replaced, its systems updated, its depth rating increased. The vehicle that descended to the Galapagos Rift in 1977 and the vehicle that makes dives today share a name and an institutional lineage, but very few original components, in the same way that a ship whose every plank has been replaced remains, in some sense, the same ship. What has remained constant is the basic function: carry people down, let them look, bring them back up. The technology of deep ocean exploration did not remain tied to crude submersibles, though. The development of remotely operated vehicles, devices that descend on long tethers and are piloted from the surface by operators watching video feeds in real time, changed the economics and the logistics of deep-sea research considerably. A remotely operated vehicle does not need to be sized around a human body. It does not need to carry life support. It can remain at depth for as long as its power supply and the patience of the surface crew allow, which is sometimes many hours. Its cameras can be positioned and repositioned with mechanical arms. It does not need to surface if the weather changes. Noah's Deep Discoverer, one of the most capable remotely operated vehicles currently in operation, can descend to depths exceeding 6,500 meters and transmit high-definition video in real time to the ship above. The ship above can transmit that video via satellite to laboratories and institutions on shore. Scientists who are physically sitting in offices in California or Massachusetts can watch in essentially real time A remotely operated vehicle moving along an unexplored section of the deep sea floor, observing creatures, directing the pilots, calling out to collect samples from a depth they themselves are nowhere near. This mode of exploration has a formal name, telepresence. It is a word that carries with it the specific quality of being somewhere without being there, of occupying a place through an instrument rather than through a body. Telepresence has expanded the reach of deep sea science considerably. A single research cruise can now provide live feeds to dozens of scientists watching from shore rather than limiting observation to the 2 or 3 people who would fit in the sphere of a crude submersible. Questions can be asked in real time by people who are sitting at sea level, and the vehicle at depth can respond to those questions within seconds. A coral colony that one scientist notices on the video feed can be flagged and approached before the current carries the vehicle past. A creature that no one in the room has seen before can, in the span of a single dive, be filmed, documented, and sent to a specialist on another continent who watches the footage on the same day it was collected. The footage that these vehicles collect is often the first time a particular patch of ocean floor has ever been seen. It is a specific quality of deep-sea exploration that discoveries of this kind are routine, not in the sense of being unremarkable, but in the sense that the seafloor being explored is largely unexplored, and so almost any location a vehicle descends into is in some practical sense a new place. New species are collected on a regular basis from deep-sea research cruises. New geological features are documented. Behaviors observed in collected specimens that have never been seen before are noted, filmed, and added to the record. The acoustic technology that builds the maps underlying all of this exploration has its own history and its own logic. Early ocean depth measurements were made by lowering a weighted line over the side of a ship and recording how much rope had paid out when it hit bottom. This process is called wire sounding, and it was slow, a single measurement at a single point, requiring the ship to stop, lower the line, and wait. The global picture of ocean depth that existed at the end of the 19th century was built from these individual soundings. Thousands of them, each representing a ship stopped at a specific location, a weight descending through cold water, and a number recorded in a log. The picture that emerged from this process was a rough sketch, accurate in broad outline, empty in detail. Single-beam sonar, developed through the 20th century, improved on wire sounding by replacing the physical line with a pulse of acoustic energy sent downward from the ship's hull, and timed on its return. A single-beam sonar system emits one pulse and receives one reflection, generating one depth measurement per ping, but it does this continuously while the ship is moving, producing a long line of depth readings along the ship's track. The picture of the seafloor that emerges from single-beam sonar is still essentially a series of lines, the ship's course traced across the ocean, and the depth recorded along it, with everything between the lines unknown. Multi-beam sonar systems change this by emitting a fan of acoustic pulses rather than a single one. A modern multi-beam sonar sweeps a wide swath of the seafloor beneath and to the sides of the ship, receiving hundreds of simultaneous depth measurements per pulse and assembling them into a continuous, three-dimensional image of the seafloor below. A ship equipped with multi-beam sonar crossing an unmapped section of ocean resolves the seafloor in real time on a screen in the mapping laboratory. Ridges and valleys taking shape, seamounts rising from the surrounding plain. The topography of a place that may never have been seen before, emerging feature by feature from the returning sound. The maps produced by multi-beam sonar are detailed enough to navigate by, detailed enough to identify geological structures, and plan the descent of a remotely operated vehicle with some confidence of what the vehicle will encounter. They are, by some measures, among the most precisely rendered maps of any surface on Earth, and yet the majority of the ocean floor does not have one. As of the most recently published assessments, somewhere between 80 and 85% of the world's ocean floor has not been mapped at high resolution. The exact figure depends on what resolution is counted as sufficient, but the broad conclusion does not change much depending on the threshold chosen. The ocean floor is enormous. It covers roughly 70% of Earth's total surface area, and the ships equipped to map it, moving as fast as they can while their sonar systems sweep the seafloor below, would need many decades of continuous surveying to cover it all. Some portions of the deep ocean are so remote from shipping lanes and research ports that no vessel has ever had occasion to pass over them with the right equipment operating. The implication of this is a particular kind of map. Humanity currently has more detailed maps of the surfaces of the Moon, of Mars, and of Venus than of the floor of its own ocean. The Moon has been mapped in high resolution by orbital radar systems. Mars has been photographed from orbit with instruments that can resolve features a few meters across. Venus, shrouded in cloud, has been mapped by radar with sufficient detail to locate mountain ranges and volcanic plains across its entire surface. The ocean floor, sitting beneath water that no spacecraft can see through and that no satellite radar can penetrate, has not been similarly surveyed. What exists for most of the deep ocean is a low-resolution model derived from satellite measurements of sea surface height, a surface that is very slightly warped by the gravitational influence of large features below, and from which the presence of a mountain range or a trench can be inferred in broad outline, but not traced with precision. This does not mean the ocean is featureless. What the low-resolution models show is already remarkable: a global mid-ocean ridge system, tens of thousands of kilometers long, abyssal plains broader than any continental landmass, trenches descending deeper than the tallest mountains rise above sea level, and seamounts rising 2 and 3 kilometers from the surrounding floor, thousands of them, perhaps tens of thousands, their exact number not fully known. The ocean floor is one of the most varied and geologically active surfaces on Earth. It is just mostly unseen. There is a particular quality to that unobserved existence. The seamounts in the unmapped portions of the ocean floor rise and are eroded and are built again by the slow movement of tectonic plates over millions of years, whether or not any instrument has ever swept a beam of sound across their slopes. The sediment on the abyssal plains accumulates, slowly, at rates of millimeters over thousands of years, regardless of whether any research vessel has ever passed above. Cold water moves in deep currents through channels between submarine ridges that have no names yet in any chart. Creatures live in communities along those ridges and on those plains and in the water above them, doing what creatures do: feeding, moving, reproducing, in the dark, in the cold, in the pressure, without any contact with the surface world at all. The scientists who work in deep-sea research often speak of this in a specific way, not with frustration at what remains unknown, but with a kind of steady interest in the fact that the unknown is right here, not on another planet, not in another time, but beneath the same ocean that has been traversed by ships for millennia. The Pacific Ocean has been crossed by sailing vessels for thousands of years. Trade routes have run across its surface for centuries. Its islands have been settled and resettled, its currents used for navigation, its surface weather mapped and studied and charted in extraordinary detail. And beneath all of that, beneath the shipping lanes and the fishing grounds and the cable routes and the Navy exercise zones, The deep ocean floor has gone largely unexamined, going about its slow geological and biological work with no particular awareness of the activity at the surface above. In a practical sense, the unmapped portions of the ocean floor are simply places where the right ship with the right equipment has not yet gone. The technology to map them exists. The ships capable of carrying that technology exist. The scientists who would use the resulting maps productively exist, and in numbers large enough to absorb a great deal of new information. What has been lacking mostly is time and coverage, the patient, methodical crossing and recrossing of the deep ocean's surface that would eventually produce a complete picture. An international effort called Seabed 2030, coordinated by the General Bathymetric Chart of the Oceans, is working toward mapping the entire ocean floor at high resolution by the year 2030. It is an ambitious target. Progress is real. The percentage of the ocean floor mapped at sufficient resolution grows each year as ships report their data and add it to the global archive. What will be found in the portions not yet mapped is by definition unknown, but the history of deep sea exploration offers some guidance about the texture of that unknown. Every new section of ocean floor that has been examined in detail has produced something that was not anticipated from the low-resolution model that preceded it. The hydrothermal vents of the Galapagos Rift were not predicted. The cold seeps that were discovered later, places where methane and hydrogen sulfide bubble up through the sediment of the continental margins, supporting communities of organisms similar in structure to vent communities, but dependent on a different chemistry, were not predicted. The seamounts that have been surveyed in detail have turned out to host biological communities far richer than the surrounding abyssal plain, their hard substrates providing attachment points for corals and sponges that would otherwise have no surface to grow on, their slopes producing upwellings that carry nutrients from depth. Each time the resolution has improved, the complexity has increased. Each new look has found something that was not visible from the previous level of detail. This is not a guarantee that the remaining 80% of the ocean floor contains wonders waiting to be found. It is an honest statement of what the record shows so far, that the deep ocean, wherever it has been examined carefully, has turned out to contain more than a coarse map suggested. The pattern is consistent enough to carry a reasonable expectation. The researchers who descend to the deep ocean in submersibles or pilot remotely operated vehicles from ship decks often describe a specific shift in their understanding of the ocean that comes from direct observation, as opposed to working from data collected remotely. A sonar map shows the shape of the seafloor in 3 dimensions, but it does not show the texture of the sediment, or the color of the rock, or the density of the biological communities living on the slope of a particular ridge. A net dragged through the water column collects whatever swims into it, but it does not show how those animals are distributed in the water, whether they are clustered or spread, whether they are oriented toward a current or away from it. The image from a camera moving slowly along the seafloor at the end of a fiber optic tether carries information of a different kind, not more precise in any numerical sense, but richer in the specific way that observation is richer than measurement. Jacques Picard, who looked through the viewport of the Trieste at the floor of the Challenger Deep and saw a flatfish resting on the sediment, could not measure the flatfish. He could not collect it. He could not assess its physiology, or its diet, or the density of the population it belonged to. He could only observe it, for the duration of the 20 minutes he had on the bottom. What he observed was that a fish was there, that at the deepest point on the surface of the earth, in conditions that would have seemed before the dive like conditions under which no vertebrate could reasonably be expected to live, Something was living. This was the kind of observation that could not have been produced by any instrument that did not look. A pressure sensor at that depth would have returned a number. A temperature probe would have returned another number. A baited trap might have caught something, or might not. The window made it possible to simply see. The deep ocean has been opened, incrementally, by a succession of windows like that one. Some of them are literal, the viewports of the Trieste and Alvin, and the dozens of other crude submersibles that have made dives over the decades. Some of them are the camera systems of remotely operated vehicles, transmitting their images up fiber optic tethers to screens aboard surface ships, and from there to screens on shore. Some of them are the slanted sonar beams of multi-beam systems sweeping the seafloor below a transiting ship, resolving a new ridge or a new depression in the abyssal plain in the fraction of a second it takes for sound to travel down and back. Each window has opened a part of the ocean that had not been opened before. Together, they have produced the picture of the deep ocean that currently exists, which is a picture of approximately 15 to 20% of the total area, examined at sufficient resolution to reveal most of what is there. The other 80% remains. Not in any sense of crisis, not as a gap demanding urgent attention, but as the simple condition of a very large place that has not yet been fully examined. The Mariana Trench has been mapped along portions of its length. Its deepest point, the Challenger Deep, has been visited by a handful of crude submersibles and by remotely operated vehicles. Its walls have been imaged and its sediments sampled, and yet large sections of the trench remain poorly mapped, and the biological surveys that have been conducted have sampled a very small fraction of the environments that exist along its length. What is known about it is genuinely interesting. What is not known about it is considerably larger than what is known. The same is true of the Tonga Trench in the southwestern Pacific, and the Philippine Trench to its north, and the Kermadec Trench running northeast of New Zealand. Each of these trenches descends below 6,000 meters. Each hosts biological communities of amphipods and snailfish and bacteria adapted to the pressure and the cold and the near-total isolation of environments that receive very little organic matter from above and have no connection to the conditions of the surface world except through the slow drift of particles downward and the long residence time of the water itself. These communities have been sampled in a general way, but the detailed picture of life in any individual trench is incomplete, and what completeness means in an environment this large and this varied and this difficult to reach is, itself, an open question. The snailfish in the deep trenches have been studied with increasing interest in the past 2 decades, as the remotely operated vehicles capable of reaching them have improved. They are not dramatic animals. They are pale and soft-bodied, with no scales, their skulls open, and their bones reduced to the minimum required to hold their bodies together under pressure. They are the deepest living vertebrates. Known specimens have been filmed at depths exceeding 8,000 meters in both the Mariana and the Izu Ogasawara trenches. They move along the seafloor and in the water column above it with a kind of unhurried efficiency, and they gather in numbers at baited camera traps in a way that suggests they are neither rare nor evenly distributed, but clustered around food sources in an environment where food is scarce and arrives unpredictably. What connects these isolated populations of trench snailfish to each other, or whether they are isolated from each other at all, is not fully known. The trenches themselves are separated by abyssal plain, and whether a snailfish could cross from one trench to another across the floor between them is an open question. These are the kinds of questions that accumulate when a place is large and mostly unexamined. They are not urgent questions in most practical senses. The snailfish in the Tonga Trench will continue their existence whether or not anyone determines how connected they are to the snailfish in the Kermadec Trench. The cold water on the abyssal plain will continue its slow movement through deep channels, regardless of whether any multi-beam sonar has yet resolved those channels with sufficient detail to name them. The seamounts in the unmapped portions of the Pacific will continue to accumulate their slow communities of corals and sponges and the fish that graze among them, exactly as they have been doing since long before any ship passed over that part of the ocean with any instrument at all. What the scientists who work in deep sea research have built over the decades since the Trieste descended and Alvin was built and the first remotely operated vehicles were lowered on their tethers into the dark is a picture of a place that continues to exceed any picture that has been made of it. Every census finds more species than the previous one. Every new section of seafloor examined at high resolution turns out to have more topographic complexity than the low-resolution model suggested. Every new deployment of a camera system in water that has not been filmed before returns footage of at least something that has not been seen before, and often something that was not expected. This is not a temporary situation, not a gap that will be closed by any single expedition or any single technological development, but a steady condition of a planet whose ocean is very large and whose surface is less patient with the difficulty of examining it than the ocean itself is with being examined. The ocean does not wait to be known. It simply continues, cold and dark at depth, moving in its slow currents, maintaining its pressure against whatever enters it, hosting its communities of organisms in each of the conditions it offers, from the warm and sunlit surface to the near-freezing dark of the abyssal plain, to the extreme pressure and near isolation of the deepest trenches. A parcel of North Atlantic deep water, dense and cold, sinking from the surface in winter and beginning its long transit through the deep basins of the world ocean, will not complete its circuit for centuries. The sediment accumulating on the floor of the abyssal plain, one thin layer representing thousands of years of slow organic fall, will continue to build long after any present-day research program has concluded. The snailfish in the Challenger Deep, if there are snailfish in the Challenger Deep, will continue to move along the sediment in their unhurried way at a depth that no human had visited before 1960, and which very few humans have visited since. And the 80% of the ocean floor that has not yet been mapped at high resolution will continue to exist in its full topographic complexity, ridges and seamounts and plains and channels, and the slow, cold communities of organisms that live among them, entirely unperturbed by the state of the maps made at the surface above. The water above those unmapped ridges is the same water that moves everywhere else in the deep ocean, old, cold, heavy with dissolved minerals accumulated over long residence times, and carrying in its chemistry a record of the surface conditions that existed when it last saw sunlight, centuries or millennia ago. The ocean at that depth is a quiet place. Not silent, there is sound at depth, the low frequencies of distant shipping and biological noise, and the groaning of tectonic plates, but quiet in the sense of being far removed from the rhythms of the surface world and from the light and weather that drive them. No storm at the surface reaches 4,000 meters in any form that matters to the biology below. No tide raises and lowers the deep ocean floor. No season turns in the bathypelagic dark. The water temperature at those depths varies by fractions of a degree over years, and the communities that live there have adjusted their biological rhythms to a world without the reliable daily and seasonal cues that organize life at the surface. What persists in the deep ocean is time, more of it, and slower than the surface world knows how to accommodate. The whale fall that descends to the abyssal plain becomes a community. It feeds scavengers for months, then specialists that consume what the scavengers leave, then organisms that metabolize the lipids in the bones over years, and finally, after a decade or more, becomes part of the sediment that underlies everything else.